as soon as. Okay. Great. So welcome everybody to this day of lectures. So before we start, I want to remind a few rules on how to interact with the speakers to ask questions. So if you are following from YouTube, you can ask questions in the chat. If you are following from Zoom, you can either ask questions. I mean, I know you are familiar with this, but it's always useful to repeat them. You can either ask questions in the chat or use the raise hand button of Zoom. So I think we can start with the first lecture. So it's my pleasure to introduce Sandro Azzaele. Sandro is a professor at the University of Padova and is broadly interested in mathematical modeling of biological systems and has given many contributions to the interface between stochastic processes and ecological dynamics. And today is giving lectures on community patterns and upscaling. So thank you very much, Sandro, for being with us, and please start when you are ready.
Okay, thank you, Jacopo. Let me. Okay, can you see my screen?
Yes, it works.
Okay. Okay, I think that we can start whenever you want. Okay, good. Okay, thank you very much, Jacopo, for taking care of this and for this nice invitation. Welcome everybody to this second lecture. This is a second lecture out of 3. The first one was given by Amos Maritain. The third one will be given by Samir Suveis tomorrow. The first one was given by Amos on let's say, an independent, model-independent approach to community patterns. Samir and myself are going to give instead a lecture, 2 lectures on model-dependent approach. In particular, I'm going to say something about neutral theory and upscaling biodiversity, and Samir is going to tell us something more about community patterns in consumer resource models. So this is kind of the plan of the 3 lectures. So why, let me give you a bit of a motivation here. Why are we going to care about modeling community patterns? So in here, we would like, let's say, to understand how these communities, how these patterns that we observe in communities emerge from underlying rules. So we want to understand, for example, whether they are stable, why they are complex, and in what sense they are complex, and if there is a relation between complexity of these communities and stability of their status. This is a long-standing problem, but it's important to understand to a certain extent to what degree these patterns that we observe are the result of a contingency to a certain extent or universality. So what we observe is something that is universal, that it affects all pattern that we observe, or is it something that strongly depends on the detail of the underlying individuals, or underlying components of these patterns? So this is, let's say, the theoretical side on one hand, but there is also let's say, a practical side, an applied side. So we would like to understand how ecosystems react when they are perturbed, and whether we can come up with some conservation strategies to improve their status. it's important to highlight also the fact that prediction here, we would like to understand the mechanisms underlying these patterns on one hand to make predictions, but it's also possible to make predictions without understanding, although this is not always safe. But you know, we live in the era of the data deluge, and if you have a good algorithm which can learn from the data, then you can infer some properties and you can make some predictions. But this is not what I'm going to do today, although it's an interesting side of this study. So another important thing to understand is that the mechanisms that we would like to understand usually operate at different scales with respect to the scales of the patterns which we observe. This is an important point. For example, when we observe patterns, we have to introduce some windows of observations, and these windows introduce some biases. For example, we could have some finite size effects, some spurious correlations, or some sampling effects. And this is, it is important that we are aware of these problems. In general, our standpoint is that community patterns in general emerge because they are the result of a collective behavior of interacting units. And from these patterns, we would like to identify key mechanisms which, from which we can derive this macroscopic pattern. All this is a fantastic challenge, but what I've just said, the fact that mechanisms may vary across scales and they reverberate on scales of observation suggest an important point. We have to be aware that when we observe the world, we are wearing a kind of a thick pair of glasses, okay? So it may well be that what we observe is not something very interesting. Let me be a bit clearer about this. So the first form of deforming lengths is randomness, like I've written, you can read here. So it may be that what we observe, the patterns that we measure, are somehow basically are not the result of mechanism. It might be that they are not the result of mechanism, but simply a result of random associations. This is a kind of a problem to a certain extent, because of course, when we measure something and we want to infer the mechanism, we would like to, we many times assume that there are mechanisms by which we obtain these patterns. However, this is not always the case. And there is an interesting paper here by Mazzolini et al. Who highlighted this problem and showed that basically we have to take care of this problem because, for example, complex systems, many of them are modular, which means that they are made out of components like Lego bricks. With Lego bricks, we can do Lego sets, and from this, because we have a list of instructions, we can come up with a Lego set, and because we have let's say a target architecture, right? So in this case, it's clear that the Lego bricks, and on one hand, and the Lego sets, the target architecture, there is a strong link between the two. However, in general, when we have functional, when we have ecosystems, usually it's not clear what kind of functional design or constraints are in place. So these are not clear a priori, right? So, because, for example, we could have some components that are like in the Lego sets, we have some bricks that are small. Usually we have very many of them, but some other which are larger, we have just a few of them. However, when we combine them, the number of these different Lego bricks are different, and this introduces some heterogeneity in the system. And this heterogeneity in the system makes some combinations more likely than others. It's just a kind of an entropy effect, which we must be aware of. So, because and this is a problem not only that is not only a problem that is present in ecology, but also, for example, in bacterial genomes or in linguistics when we analyze book chapters and many other patterns. For example, there are situations in which you can have horizontal gene transfer in which the randomness is present, but there are also other components that are not as important as this, or the other way around. So we have to make sure that what we observe is important, is driven by an underlying mechanism. So how do we do that? Basically, the idea is to build up some null model, and now we are going to see one of them, some null models from which we can see, so basically we can derive from whether they work or not. So basically these null models are informative when they fail. This is the basic idea. When they fail, we see that probably there is a mechanism, there is something underlying the pattern that is probably interesting. So, and this tells us whether there are specific architectural features in our patterns. So now let me start with this kind of introduction, long introduction. Let me start with the first pattern here, which is probably one of the, is very well known probably to many of you here. So it's the species area relationship. Here, this gives you just the mean number of species as a function of the area. Okay, and this is very well studied by ecologists. We know very many things about this. But let me start the idea, and this is something that I think should be clarified, like I said, not only in ecology, but in many other areas. So here, let me give you an example. We have a set of islands, here the Galapagos Islands, and here we have the area, and in each and every area, we count the number of species. So here, we are not looking at populations, just number of species. Okay, if you put on the x-axis the log of the area and on the y-axis the log of the number of species, you get these points here, these green spots here, and you can draw a straight line. This means that you have a power law, right? So from this, now the question is here. So can we obtain this sort of pattern by placing at random individuals in space? This is the question. This is what I had in mind before. So do we have this kind of lens, the forming lens? Is this a pattern that comes from something that, from a mechanism, from an underlying mechanism or not? Usually what ecologists do is basically they don't care about this. Sometimes, not all of them, but sometimes they don't care about this, and they say, okay, let's draw a straight line. This is just proportional to a to the power of z, where z is always smaller than 1. So always, and usually greater than .2. Okay, what happens in this case? Let's try to put at random these individuals in space. So we have species I has n I individuals within some total area, capital A, and then we place at random this over an area. Now, if we want to look at the number of individuals that fall within the area small a, we get that the probability that at least one of these individuals, individual may fall within small a is given by this expression here. So 1 minus a over capital A is just the probability that no individual falls here, and this one individual, and this is for the entire species. Now, if you do the calculation, you end up with the equation 1, with this formula 1, which tells you that okay, if we assume to know the distribution of individuals among species, okay, then the species area relation that we should get is simply given by this expression here. Now, if you want, there is a homework here. You can calculate the variance of this. So basically, you can assume that you are given a Bernoulli random variable which gets 1 when an individual species I falls within area small a, and the probability is given by this expression here, and otherwise is 0. So this gives you the average value, but you can calculate the variance as well. It's not difficult, and you can find already the answer in equation 2 of this paper here, which is very interesting, although it is quite old. So what happens when we go ahead and we try to compare this with the data? This is what we get. So if you go out there and you collect the data from the Lambir tropical forest or the Paso tropical forest, you get, I mean, the black dots here are those given from the empirical data, but the random placement model is the dashed line. So as you can see, is here and here. And this is kind of a common behavior. So basically, the idea here, as you can see, you get a systematic overestimation of the number of species at all spatial scales. So it means that somehow here, somehow, let's say, this is good news. So there is some work to be done. Somehow we see that the patterns, that the species area relationship basically has something to tell us. Something more important than just randomness. And this is also true for other patterns that we are going to see in a minute. The reason why here I've shown you that the random placement always overestimates. So the reason for this is that you can see this, you can see the reason from this picture here. These are 8 different vascular plant species in the Paso forest. You see, they don't look randomly placed in space, they look aggregated. And this is something that now basically all ecologists have understood, in the sense that randomness is one component, but is not the most important one. And usually species, vascular plant species, are aggregated in space. And if we want to model this, if we want to model spatial distribution of species, we have to take care of this aggregation in space. So, okay, let me just do a first summary. This is a first, sorry. This is a first a summary of the first part, very simple, but I would like to make things clear. So we have the species abundance, the species area relationship, and many other, which basically are not just the result of random association. We need more, so there is more work to be done here. On the other hand, this SIR is subadditive, in the sense that if we double the sampled area, then we don't double the number of species, but we get a smaller number, and this is a smaller number of species, and this is because the Z exponent that I've shown before is smaller than 1. And usually ecologists use this power law, but there is no compelling theoretical reason for that. So basically, if you write down a model, a spatial model, you can get something that is different from a power law, but in some regime is a power law. So basically, this is just a phenomenological approach. So don't worry if you come up with a model that is not a power law but fits the data, is not a big problem. And finally, what we have learned is just the species are aggregated in space. So this is a summary for the first part. Jacopo, I don't know if there is any question, or I can go ahead.
So far, I don't see any hand raised nor any question in the chat, but if you have any, please, now is the time to ask. Well, I think we can move on, and then
okay.
Thanks.
Okay, so there is, now let me move on to a second empirical pattern, which is the species abundance distribution. So in this case, we look into the abundances of species. So before we have looked just at species riches, now we look into populations and how these populations are distributed across species. The usual pattern, that the typical SAD is given by these two plots here. So on the left-hand side, you have on the y-axis, you have the number of species, and on the x-axis, you have the log2 of the number of individuals. This usually you have a peak in the middle, even though not always is as big as this, but you have a peak in the middle, and this is the reason why usually we get this kind of log normal behavior, which is used by ecologists. And here you also have this rank abundance. So this is telling you that there are just a few species with a lot of individuals, but a long tail of fewer, a long tail of many species with just a few individuals, rare species. Here there is another piece of homework. If you want, you can calculate the relation between the two, and you can see, you can realize that one is proportional to basically the inverse of the exceedance probability of the other. So if you want, I can give you the details, no problem with that. So, okay, these are two ways to present the same piece of information. So in here, let me give you a caveat here, because it's important that we understand how we collect this data. You know that in tropical forests live many different kinds of species. We have creepy crawling creatures and chirping birds and trees and whatever. Are we going to put everything in this SAD? All the species are going to be analyzed in this species abundance distribution? The answer is no, no. And the reason for this is that if we wanted to model this, it would be basically impossible to come up with something reasonable. Basically, it's too complicated, because we should calculate the probability that species 1 has n1 individuals, species 2 has n2 individuals. But if we want to model this, we have to deal with hundreds of species. All of them are interacted in a trivial way, and this interaction may vary across different spatial scales. So no way. There is currently, basically, we don't know how to model quantitatively large-scale spatial patterns in multi-trophic communities when there are many species. So the idea here is to introduce an empirical model system. So basically, we focus just on ecosystems with one trophic level. Okay, so, and in here we introduce some simplifying assumption. Here we have species of bees, overflies, species of breeding birds, and so on. So in our case, we are going to focus just on plants in a forest. When individuals within this tropical, within let's say these ecosystems with one trophic level, individuals are roughly, can be considered independent, or they interact more or less in the same way, and they are clumped, like I said before. So in this way, we can introduce some simplifying assumptions. So in here, as Hubble as Hubble did at the beginning of the millennium, the simplifying assumption that he introduced is that individuals basically of any species, of all species, interact, compete in the same way. So they all have the same probability to give birth, dying, migrating, and so on and so forth. So the identity in this case is not important, and even though it may seem that neutrality is a very strong assumption, basically we are able to introduce some models which can justify and which can tell us more about the patterns and macro scale. And although this assumption is completely false from an ecological point of view, it is very useful when we want to introduce some baseline community patterns, some kind of null models, models from which, from the deviations of which we can understand something more about the underlying mechanisms. So, okay, so if we make this assumption, then we have said that we have said that the identity is not important. So we go from P of n1 and 2ns to P of n, because we have basically one species in a given region. And here we can basically write down a master equation, which is here. This is telling you how it behaves, this probability of having a given number of species with individuals. We can write this from one step to the other. When you simplify, you get the master equation. And if B naught, which is the birth rate when there are 0 individuals, and D naught are 0, then this master equation may have a steady state, which can be calculated in the following way. I'm going quickly about these technical details, but I can leave you all the slides so you can do all the calculation by yourself. So Pn star can be calculated recursively, and you end up with this expression. Pn star is just the product of these ratios of birth and death rates. And because of a normalization, you can calculate explicitly this steady state. Now, with this in our hands, we can basically model tropical forest. The first assumption is just to say that B of n is proportional to n, and D of n is proportional to n. If you do that, you plug this into the formula that I showed you before, you get what is called the Fisher's log series, which is widespread and has been studied many times, and is in good agreement in several, let's say, in several ecosystems, although it can be improved. And the improvement is the following here. We can modify just by adding one constant, so Bn, instead of n, we have plus r and plus delta. And thanks to this new expression, we can obtain two different species abundant distributions. We can have this, which is the negative binomial, which goes back to the Fisher log series as r goes to 0, or we can have this expression here, in which when c goes to 0, we recover Fisher log series. Why two different expressions? Okay, these are good, equally good to a certain extent when it comes to model empirical patterns, but it turns out that the negative binomial has some interesting mathematical properties which are not shared by this other S-den. And we are going to see in a few slides these mathematical properties. So when we compare this model here, this model with the density dependency to the data, we get this very good agreement with the data, and this is well known since 2005, more or less. So good agreement, although this is, it doesn't imply that we have neutral mechanism at the fundamental level. We are able to show that there is some degree of universality in these patterns that we observe. Another thing that I would like to highlight is this x, the value of x, which is very close to 1. And I would like you to bear in mind this value, these values here that are close, because it will become important later on when we are going to model spatial degrees of freedom in neutral theory. So, okay, another summary now. So we have neutrality, which is at individual level, and it tells us that we have just per capita ecological equivalence among individuals. This is at individual level, not at species level, like MacArthur did in his theory of island biogeography in the 60s. Then this is the niche theory is in contrast to neutral theory, because niche theory asserts that species coexist because their identity, because they are able to partition limited resources. On the contrary, instead, neutral theory confers a lot of importance to stochasticity, to ecological drift, and basically it washes out the all species identity. And because of this, it's able to recover some, to identify some underlying universal patterns, universal mechanisms. So as you know, all models are wrong, but some are useful. And I think that neutral theory is somehow useful because it helps identifying these universal drivers. And another warning here, neutral patterns here, of course, even though we are able with neutral theory to, let's say, to match the empirical data, this doesn't mean that there are in place neutral mechanisms. This is important because there are some other mechanisms, for example, some other models which are non-neutral, like the hierarchical competitive model by Tillman, which can produce log series distribution even though they are non-neutral. But that doesn't mean that it's truer than neutral theory. Also, there may be some life history trade-offs which introduce some equalizing effect on birth and death rates, and therefore they look like more neutral. Okay, this is a second part. Jacopo, if there are questions?
No, I don't see any hand raised. Is there any question? Anyone that wants to ask something?
We can move on, no problem with that.
Everything is crystal clear.
Maybe everything's clear,
Yeah.
or everything obscure.
It's always one of the two extremes, but
Yes.
I'm optimistic and assume it's the first one.
Okay, so let's move on now. Okay, up to now we've seen the species abundance distribution and the species area relationship. Now we would like to go ahead to move on and try to link them with one approach, with one model. So on one hand, when you see you have this forest, when you sample small areas, you get this species abundance distribution with a mode that is basically at when the number of individuals is very small. But then when you increase the sampled area, this mode goes into higher classes here. And so you move from small number of species up to large number of species. This is, as you can see, this is clearly a kind of a sampling effect, which is difficult to model, and this is one of the reasons why, for example, in the upscaling problem that I'm going to explain later on, this is kind of a challenge. So patterns are scale dependent, and when we model this, we have to be careful, and we have to look into this problem. So is it possible to come up? So here I've just shown you species area relationship and species abundance distribution, but there are many other patterns that can be measured. Here is just a list. Species lifetime distribution is a pattern that involves time, pair correlation involves space, so on and so forth. Taylor's law, okay, I don't want to look into the details of this, but the idea is to find out whether there is an underlying mathematical theory from which we can derive, if not all these patterns, at least some of them. And now it's what I would like to explain you. So in here, we start with a model which is basically defined on a lattice, okay? It's a meta-community model, so in every site we put several individuals, and there is no threshold on them. So we have, and how does it work? It's just, it has 4 moves. So the first is a death move. A death move in which basically one individual here of one species in site I dies at a rate R. Then we have the birth move. So we have an individual, and this produces one offspring at rate B. And this can hop onto one of the nearest neighbor sites with probability 1 minus gamma. Finally, we have that every site is colonized by a constant rain of propagules with rate B naught. Now, it's possible to write down the birth and death rates of this model, and therefore we can also write down the spatial master equation for this model. Let me give you just highlights of the most important properties. It is species independent. There are just 4 basic mechanisms: birth, death, diffusion, and immigration. So basically in here there is only demographic stochasticity, no other effect. If you look at the mean, the behavior of the mean is quite trivial. So the patterns here emerge simply because of demographic stochasticity. There are no environmental effects, and because the birth and deaths are linear, there is no carrying capacity. So in here, B must be always smaller than D. And biodiversity, so the states, the stationary state is non-trivial when B naught is larger than 0. Because of the term that I showed you here, this sum here, this tells you that we have spatially aggregated species. And finally, this model is minimal in the sense that if you get rid of one or more of these parameters, basically you end up with something that is trivial. So with these patterns here now, I mean, this section is a bit more technical, so I will go quickly over this part. I just want to show you how it goes, but there are papers where you can find all the details that you want. So here you introduce this partition function, you write down then the equation for the partition function, where you have the partition function in discrete space. Then, because basically this equation 4 is too complicated and we don't know how to use it, then we go back to the data and we look at what kind of regime we are interested in. So what we know is that B over R, which is the X that we've seen in the first part, you remember, always the X was close to 1. So here we are going to do the same. B over R, which is the old X, is always close to 1. And D, the spatial diffusion, is always comparable to R minus B. Okay, in this, starting from this, you can introduce two different parameters, which are epsilon and eta, which are defined in this way, and you consider a regime in which both they go to 0, but in a way that the ratio is order 1, and also B over B naught is order 1. Okay, if you do the calculations, you do all what, all the proper calculation, you end up with this new equation 5 equation for the partition function, and eventually it turns out that you can introduce a new partition function for the global patterns, for the patterns, basically for the random variables that you need in order for you to calculate the species area relationship and the spatial species abundance distribution. This can be derived directly from the partition function, and you end up with this equation here, in which you have this Laplacian here of f of v, which introduces the spatial effects. So the effects, the spatial effects of what you are looking at or what you want to derive are enclosed in this term, in this function f of v. Now, to cut a long story short, basically it turns out that we know this function f of v, but the really crucial point, the really important point is that when you introduce this, when you introduce f of v into the equation, basically, miraculously, this integral of a Laplacian disappears, and you end up with this function, with this equation 6, in which you have just this term sigma, capital sigma, which is proportional to the Fano factor. And the Fano factor basically is tells you how your fluctuations deviate from a Poissonian, from Poisson fluctuations. So in this, you can really simplify things, and you can understand that basically space enters the equation only through this function here. And this function here can be solved exactly. Okay, of course, in this specific regime. So here we have a stationary distribution, which is our probability, our species abundance distribution, which depends on space only through this sigma, capital sigma function. So, okay, this, okay, if you want, you can also introduce time, but I don't want to look into this. But anyway, it's possible to look into space. Also in this regime, which is basically what is interesting from the applied point of view. So in here we have an underlying mathematical model from which we can derive basically all the patterns, so we can link them in a consistent and controlled way. Can we compare with the data? Yes, the answer is yes. And what, okay, here I'm going to show you what we've got for the Barrow Colorado Island, but we did also for other forests, and we got comparable results. So, okay, this is just the mean number of individual per species, and I think that every more or less every conceivable model can fit this data. Anyway, we can go ahead and we try to best fit now the pair correlation function, which is given by this model, by this expression here. This is the exact pair correlation function that we can calculate from the model. Okay, now we have all the parameters, we can make some predictions, and the predictions told us that basically the Fano factor is always very, very large. The correlation length is large, and the correlation time is also large. So in the regime that we are considering, the model is able to match the empirical data when is close to a critical point. So when mu is very close to 0. So large fluctuations, fluctuations are large, and they vary on large temporal scales and are correlated on large spatial scales. So now that we have the data, we have the parameters, we can make the prediction. So we have this red curve, which is a true prediction. We can say how the SIR behaves across scales. As you can see, it goes more or less on top of the data. And also we can predict how the RS, or the species abundance distribution, looks like at the global scale. And this is what we get from, like I said, this is a prediction. And also when we downscale the prediction and smaller scales, this goes more, is able to explain the data that we observe. And this is also for the passo forest, so with comparable results. So, okay, third part that we are going to summarize here. So in some regimes, we can derive analytically spatial explicit models of commutative patterns, and we can link the patterns in a consistent way. It seems this model is going to tell us that it's the model, if we want the model to match the data, we need to, let's say, we need, basically it's telling us that we are close to a critical point, okay? Although that doesn't strictly imply that we have a critical behavior in this case from the data, but okay, this is at least what the model is telling. And if we want, this model can also include environmental noise. So you can get all the patterns that we've seen when we have environmental noise. Okay, I think that this summary, with this we have the third part, which is, so if there are any questions.
Yes, there are a couple of questions in the chat. So Alfonso is asking how dependent is the model and its results on the scale of the grid?
Actually, the results that I've shown you are in a continuous approximation. So in here we used large scale. So basically there is no dependence on the spatial grid. So basically what we are going to say, what we assume here, that you cannot go to really fine scales with this, because we have taken this continuum limit in space.
And Ankit is asking, is the immigration term indicative of immigration from a source pool? If not, how is it different from the diffusion between grids?
No, the immigration is different from, because the immigration is something that comes from the outside of the system. So diffusion is telling you that basically individuals hop onto nearest neighbors, but are always belonging to the lattice. Instead, immigration is a contribution of species and individuals that come from the outside. So it's kind of a different. So let's say one is local diffusion, the other is global, because it affects all the lattice.
Yeah, he's asking another clarification about this. Given the model assumptions, do all the species go extinct after long enough times in absence of immigration?
In our assumptions, no, because we used reflecting boundary conditions. But if one wants to study this, it's possible to understand also this behavior, and the solutions that I've shown you before can account for species extinction depending on the area of starting area. But this is something that I didn't show. But it's something that can be done.
Okay. And Stefano is asking if you could provide more details on how the environmental noise can be inserted in the model.
That's a very nice question. This is something that we so basically it's difficult to explain without going into the. So there is a change of variables which helps you map one problem to the other. And so basically you can find the corresponding model by using, of course, an appropriate change of variable, which maps the problem of the environmental with environmental noise into the one that we have just shown. So basically you can go from one to the other. At least, let's say, and at least at the course level. So when we did the continuous limit approach, the continuous limit in that situation. But anyway, the patterns can be derived basically in the same fashion.
Great. I don't see other questions in the chat, nor people with the hand raised.
Okay. So, Jacopo, how much time left?
You have about 15 minutes.
Okay. Okay, so may I move on?
Sure.
Okay. Okay, let's now look into the upscaling problem, which is somehow related to what we have looked into up to now, but it's on one hand is different, and it has some interesting applied sides, which may some theoretical and applied sides. So basically, let's imagine to have the Amazon. We have in our the problem is the following. We want to let's say predict how many species we have in the Amazon forest. Okay, of course we cannot go onto the Amazon and survey everything. This is too difficult, too time consuming and resource consuming. So it's we cannot do this. So what is the strategy that we could use in order to give an estimate of the total number of species that live in the Amazon forest? Let's focus on, for example, plant species, because like I said, finding out, so let's focus on one trophic level, because the full problem is basically too difficult. So in here, the idea is to do the following, like it is suggested by this map. So in here, we scattered some fine scale samples around the Amazon forest. And when we do that, we collect all possible information within one sample and all the others. So we collect all the species identities, all species populations, so on and so forth. Now, okay, we have an incomplete set of information. We have some information. We don't have a complete information, but we have something. How can we basically link this? How can we use harness this information in order to upscale and in order to infer the total number of species? This is the upscaling problem. So basically, we collect information from fine scale samples. We want to infer, we want to make predictions about species richness at coarse scales, at very large scales. What can we do? I mean, here there is a long history of different models, and for a recent monograph, you can look into this final paper by Kunin, and you have a long list of different approaches, but more or less all of them have some problems and some. Let me go into what we suggested, starting from what I just told you now. So here, okay, there are several problems that we have to face. So the species-area relation is not additive. These species are usually spatially correlated. Instead, we used some uncorrelated distributions up to now. So there are several problems here. And one of the big problems is that there are, I mean, species populations varies across spatial scales. So basically what we see at small scales, for example, here, when we have small number of species, the species abundant distribution has one shape. But we want to predict something at much larger scale when the shape of the species abundant distribution is completely different. So this is quite challenging, and it's not easy to come up with something that is consistent and is coherent and controllable to a certain extent. However, what we have, and now I'm going back to what I told you before, we can start from the negative binomial distribution, which I showed you at the very beginning, which has this form here. So if we assume, so let's say we know that this can match the data quite well at the global scale, okay? So when we go downscale, so when we look instead of looking at the entire area, if we look at smaller scales, how does this distribution behave? So there is an important property, there is an important mathematical property, which is the following, which basically it is what we are going to use and to exploit. So basically, if we start from a given negative binomial distribution when all species are sampled, then if we go down to smaller scales, then the distribution at smaller scales is still a negative binomial, which means that it's form invariant under binomial sampling. So basically, if we can, to a certain extent, start from a rough independence assumption, so basically we assume that for the time being, let's say, spatially are more or less spatially uncorrelated, then we can use a binomial sampling. And this binomial sampling, when applied to a negative distribution, is telling us that even the smaller scales, the negative binomial remains still a negative binomial. This is a very, very nice property which can be used and can be, with this we can somehow avoid the problem that I was talking about before. So what happens? So in this case, so here, this is what you have to do when you apply the binomial sampling to your negative binomial distribution. When you do the calculation, you end up with this expression here. So it is still a negative binomial, but now, as you can see, there are two interesting features. R, one of the two parameters, does not change across scales. So it is an invariant, a scale invariant parameter, but X changes with scale, and you get this X hat, okay? But this X hat now can be inverted, so now you can get X, which is the parameter at the global scale, as a function of the parameters calculated at smaller scales. So this is telling us the following interesting suggestion. So here we can, when we sample our area with fine scales samples, we can measure this X hat and P and R. And then by using this formula, which can be deduced straight away from the approach that I've just shown, you can upscale and basically say what's going on when, so basically you measure SP, which is the number of species at scale P, and by measuring X hat, and then X as a function that is outlined here, you can infer the total number of species. More details are here, but basically this is the idea. And this, we can do this simply because of this nice property of the negative binomial. By using this, we can infer, for example, the total number of species in the Amazon forest, which is kind of a modifies previous estimations here. And here there is a list, and now we can go, if you want, I can explain what we did here. But anyway, it's possible to find out how many species, at least in some specific ecosystem with one trophic level, what kind of distributions and what kind of predictions we have. Of course, we have also the species abundance distribution. So when instead we do have some correlations, then we have to use a kind of more phenomenological approach, but still working. And this just going quickly through this model, to this approach, is the following. So we start from a given SAD, species abundance distribution. In this case, we can pick a, let's say, a two parameters SAD. Then from this, we can pick another we can pick another spatial correlation here with 2 parameters as well. And from this, we can calculate the spatial variance here. At this point, we can make a link. So we substitute to alpha and beta here, 2 functions, alpha of r and beta of r, and we set these coupled equations, integral functional equations actually. So in here you impose that these equations, self-consistent equations, satisfy these 2 equations, and these are equations for alpha of r and beta of r. It turns out that in some situations, alpha of r and beta of r can be found exactly, and when you have this, you can make some predictions. How does it work? I mean, it works fine. And here you have an application to the breeding birds. Here you have some more or less 1,000 plots in France. You apply this machinery, so you try to best fit the data with this function, but you can use another function, it doesn't matter. You can do it again, you can do it anyway. And then you can predict. So here you have more than 3 orders of magnitude here, and we in France, I have been told that there are more or less 300 species expected, even though I didn't find official predictions. And with this model, we predicted 330 species by taking into account this kind of correlations. So, okay, this is the upscaling model. This is the upscaling approach, and I'm going to finish now. So we have this modeling community. This has interesting theoretical aspects which are related to the hyperdominance of species, which is still an open question. If we want, we can go into the details of this. We can also, if there are spatial correlations, if there are not, we can use the negative binomial distribution, which is robust under scale variation. So, and this is because we have this form invariance. And in any case, if we don't have this advantage, and we do have spatial correlations, and these correlations are important, it's still possible to make some predictions with a more phenomenological approach, and we can predict the species abundance distribution and the species area relation. This approach can tell us basically not only how many species we are missing, but also what is the distribution of populations among species that are yet to be observed. Okay, I think that I'm done, and I just wanted to thank you everyone. Here is our lab, the LEAF lab, and here are the collaborators. Of course, what I presented is the result of deep interaction with all people, so I'm in debt with all these people without whom I couldn't get anything of what I presented. Thank you very much to everyone, and thank you, Jacopo.
Thank you very much, Sandro, for the very nice lecture and overview of these results. So there is time for a few questions. So please, if you have any, raise hand or write it in the chat. Well, I mean, since no one is asking, I have a question myself. So, oh, actually there is one, so I'll give priority. So Silvia is asking, does the change of the X parameter under rescaling of the negative binomial distribution reproduce the different shapes of the SAD at different scales?
Yes. In fact, it goes from one mode, from one, let's say, internal mode to the distribution where there are no internal modes. Basically from internal peak when we have the negative binomial behavior to the Fisher log series behavior when the mode is always in the first class.
Great. Any other question? Well, then I asked the question I wanted to ask. So one thing that it's surprising for me is that it seems that the same distribution hold across several orders of magnitude in scale, and somehow it's believed.
Are you referring to the upscaling problem?
Yes.
Yeah.
Yes. Somehow, I mean, these the negative binomial is at least partially justified mechanistically with
Yes.
neutral models, right? And I mean, it's sort of believed that neutral models or neutral theory works well at the local community scale because individuals are relatively few and there is and demographic stochasticity is more important. So I wonder, I mean, the fact that it seems that it works also for much larger scale.
No, but you see here, actually, probably I should have been more, I should have been clearer. When I address the upscaling problem, I'm not making any assumption about species. So in the first part, it was model dependent and I assume neutrality. But in the second part, when I explain the upscaling problem, in that case, I just use the negative binomial. So I'm not saying that I'm using the negative binomial because I'm assuming neutral theory. This is just I'm just using a distribution because of its nice mathematical properties. I could have derived this from a non-neutral model or whatever. We are using the negative binomial simply because it has this form invariant. So there are no assumptions in using that. So that's probably answers your question, or I don't know, partially.
So the reason why it works is because of the flexibility of the negative
Yes,
binomial.
yes, that's the point. That's the point. It's very flexible. That's the point.
Thanks. Any other question?
Bert?
Bert. Are there obvious examples of when the continual approximation fails, when discreteness is important?
So this is a question for the spatial model.
I guess so.
Yes. Okay. Here there are two things that there are two continuous limits. One continuous limit is referring to the basically to the individuals. So this is one continuous limit. The other continuous limit is the continuous limit in space. So the approximation, the discreteness of population is less important than the other somehow. When you use the continuum limit in space, yes, there is a limit. Basically, when you go down to scales as small as, let's say, compared to meters or tens of meters, basically this doesn't work very well. So basically, when you go down to scales that are much less, much smaller than the correlation length, then there are differences, and basically you cannot use the approximation that we used. But anyway, this is a model which was devised for large scale, for macro scale patterns, not for small scale patterns.
Great. So Bert is satisfied. So if there are no other questions, thank you, Sandro, very much again for giving this lecture. So now we are moving for
Hello, Matteo.
Hi, Mercedes. How are you doing?
I'm fine. I see you are going to be chairing this.
Yes, unfortunately, I'm not very lucky.
Why? Why? No, I mean, Jacopo, Jacopo is good at this, but anyhow, we'll see how you manage.
So, yes, we wait for, yeah?
Massimo,
Yeah.
may I close the breaking room? Because it takes
Yes.
it takes 1 minute. Okay.
Okay.
Starting from now in 1 minute. Okay.
Thank you. Okay.
Yeah, I think I only have to share my screen when everybody is back.
Yes, we are waiting just a couple more minutes, or one more minute for the other, for all the participants to join again.
It's interesting, the breakout groups. I've been at an international meeting where they did a lot of stuff a priori with, people kind of choosing whom they wanted to perhaps get to meet and make appointments. In the end, people, because, being online, listening and listening takes a lot of, so people in general, right, didn't come to the breakout groups. So I don't know how that works, but in that meeting, it was a bit like that. Anyhow,
So I think probably we can start, and
Yeah.
we are going to, for the second lecture of Mercedes Pascual, we are going to learn about this hyperdiversity that we are all very interested in. So Mercedes, can you please share the screen?
Yes, one moment. Okay. Do you see my first slide?
Yes.
Okay. Let me just see this here. Okay. So, okay, nice to be back with you. And yes, this will be more of a research talk relying on what I told you last time. But first, I wanted to acknowledge my main collaborators who did a lot of this work, my previous postdoc, Shai Pilozov, who now has a position back in Israel, and then Shixin He, who will soon be part of the faculty at Purdue University. So yes, today I wanted to touch on assembly or the interaction of ecology and evolution, but in a system, as I said, with a lot of pieces. And I put here a second arrow coming back from the assembled system back to the diversity of the building blocks, which is a topic that I like to touch upon towards the end of the lecture. And you have seen this plot more times than you probably would like. I just wanted to remind you that for the purpose of the parasites, we will be talking about niche differences, differences in traits here that confer an advantage as a function of frequency, so an advantage to the rare, a disadvantage to the common. And in pathogens in general, we have the advantage, I think, relative to plant systems, because we do know which traits belong to this axis. In particular, that the traits that essentially relate to recognition by the immune system and the host gaining information, memory about the pathogen, belong here. So that the distance or the overlap between pathogens in in the traits, in the molecules that are recognized by the immune system, the antigens or epitopes, that's what matters to this difference because of cross-immunity and cross-protection and access to hosts. Now, of course, I recognize that some of you, of course, are not so interested in pathogens. I like to ask you to make the effort to think that everything I say, I think could be extrapolated. Of course, there are issues of level of organization because I'm going to talk about variation within a species. But I like to remind people that a very interesting and I think a very important hypothesis on diversity in rainforests, in coral reefs, the Johnson-Connell hypothesis is basically a mechanism that relies on frequency-dependent interactions with natural enemies. These are specialized interactions with natural enemies that act against your own offspring close to you. And I'm not going to describe this in detail, just to point out that these are important mechanisms. There is a lot of recent literature on evidence for this kind of force. And I think it's a fascinating topic. Of course, we have the problem, and someone yesterday raised the issue of mutualism. This is a recent paper for these kinds of interactions in the rainforest, these Johnson-Connell mechanisms, but also bringing in mutualists. I remember someone asking about that yesterday. Of course, finding evidence of a particular mechanism doesn't mean that at the macroscopic level of the whole community or the whole system, they are important in the patterns of diversity. So we also have a problem in these very diverse systems in asking how should we determine whether those patterns matter. And of course, the purpose of neutral theory was to help in that respect. Now, I'm going to be talking about a system here, a parasite, the main parasite of malaria, and in particular about this parasite in very high transmission regions. These are the endemic regions where there is very high prevalence, in fact, very high asymptomatic prevalence, because people gain protection against the parasite, but continue getting infected through life. So in some places in West Africa with very high transmission, you find prevalences in, and I don't mean clinical prevalence, I mean prevalence in the population that can be 40, 60, 70% of the population carrying the parasite. Of course, this is a very large reservoir for transmission. So in essence, we can think that the challenge of elimination and control in these regions is related to this very high strain diversity. So can we try, in this case, to understand diversity, not for the purposes of persistence, but for the purpose of elimination? And for those of you who do not come from within biology, most people know that malaria is vector-borne transmitted, but importantly, it has a stage in the blood, that's the asexual state, and then a sexual state of the parasite in the mosquito. Now, this is what I like to do today. I like to show you that in this system, which is, I will show you, hyper-diverse from the perspective of antigenic variation, negative frequency-dependent selection, because of competition for hosts, is really acting. This is based on some theory. This is computational theory based on stochastic agent-based models and network analysis of the structure of diversity that emerges from this assembly. I like to touch a little bit on consequences of this structure for resilience to perturbations, on consequences for the existence of a threshold in diversification that I think is important, because below this threshold, systems will possibly persist in a more fragile state. So just to start, we heard about multidimensional trait spaces in terms of niche differences. Yesterday, we also heard Daniel Fisher, and we will hear more about essentially evolution in these very, very large spaces. Here is one, and parasites and biology do it in a combinatorial way. For the very specific purposes here of our system, our parasite has 40 to 60 genes that encode for the same kind of protein. This is the major antigen seen by the immune system. So for our purposes, the phenotype of interest will be a combination of 40 or 60 genes. Now, if you ask these genes that encode for this antigen, how many different genes we find in a local population in these high transmission regions of the order of tens of thousands. So you can do the combinatorics. Of course, it's astronomical. And so what the immune system sees and gains information on is this very heterogeneous population of strains. Of course, this molecule, I just will say a little bit about it. It's this molecule PFEMPA1. It's basically exported to the surface of the red blood cell because it has a function. It makes the red blood cell sticky. It helps the parasite stick to the walls of the capillaries, etc. I will not worry about function. I will just say that because it is so exposed, it's very involved in immune evasion. And so this is what the parasite does. Once it infects a red blood cell, it expresses one of these genes, then it expresses another, then it expresses another, and believe it or not, there is a certain synchrony of the whole population inside your infection. So they are showing their cards to the host in a sequential fashion. What this achieves is a longer infection than if we were showing everything at once about the phenotype. So our fitness here will be affected by duration of infection. Now, because if a host has gained memory, the parasite does not express it. So you have a shorter infection, shorter growth rate of the parasite, etc. So that's fitness. But if you, as I said, you could think of any other way in which the fitness is affected, doesn't matter. Here, it is duration. And on top of that, what the parasite plays a game of incredible diversification, because different phenotypes, different repertoires can shuffle their genes during the sexual phase in the mosquito. And during the asexual one, they can produce true innovation because different genes can produce new genes. So all this variation, and there is very high, of course, if there is high transmission, there is high recombination. And so you can say, well, there cannot be structure to this mess. There is this huge number of genes, new genes, recombination of the combinations is their structure. So this is work in collaboration with my colleague Karen Day, who is a molecular epidemiologist, a phenomenal one working on malaria. And she basically sequenced the marker of these genes in every child in these populations from Gabon. And what you see in this matrix here in blue is basically all the genes isolated from each child in each row against these same isolates. And then you ask with this pairwise type sharing, how much overlap there is between these. All you have to see is that there is a light color. So essentially, each infection is almost unique. I mean by that, each combination of these antigens that the hosts are seeing with an infection are almost unique. You can tell me, well, that's not surprising. If it were a random system, there is so much variation, we expect this. But if you randomize appropriately, keeping the frequencies here on the right on top, you see that the degree of overlap here in red, the mean overlap, is lower than the one that you expect at random. So there is a force pulling these things apart. So we will ask whether this is frequency-dependent competition, and if so, how can we tell? I mean, how should I look at this data to be able to tell? Now, from my previous lecture and a bit of art, you may expect that we should look for clusters, that the emergent niches from these kinds of interactions should be clusters of similar parasites. This is not exactly what we have. So what should we expect to see? So existing strain theory, as I showed you last time, will not encompass this very, very large empirical diversity. It also does not necessarily in some forms allow for, at least in the versions for malaria, for true innovation. What I mean by that is a system whose pool is open to new variation. And mostly there is no explicit comparison to neutral models. By that I mean models that retain the demography of transmission, all the birth-death processes, the extinction, but have no specific interaction. So we built, we extended, in fact, an individual-based formulation we had from the past, in which we basically have a pool of var genes from which we get immigration into the system. Sorry, we start assembling the system. We have transmission between hosts, and each gene is composed of 2 variants. This is needed here because we need to be able to have recombination generating new genes, but you could do it in a different way. So what we have is a system in which each host remembers all the I'm calling, sorry, this is advancing by itself. Each host remembers the types, the genes it has been seen before. I will use the word genes, but I mean their product here, really. And if they have seen something, then the parasite cannot express it. So they have shorter infections, and essentially that parasite will transmit less. So we basically have the mitotic recombination, the meiotic recombination. We can do that in the computer. And we can also build neutral models. We build 2 models here on the right. We basically have a model in which transmission occurs, but there is no, essentially no specific memory. There is just complete neutrality of parasites coming in and infecting the host. The one in the middle is more interesting epidemiologically, and it's what models typically do. They say, well, your degree of protection is not a function of whom infecting you, but how many times you have been infected. So this has a protection, but again, it's a generalized protection. And we parameterize these neutral models to match the duration of infection with age, so that we are not comparing apples and oranges. What I wanted to tell you is we basically look at which properties will give away that there is a structure, and that there is a structure in which this competition for hosts is playing a role. So for this, we decided to look at networks, because with the recombination in the system, we cannot look at trees. And I think these networks are appropriate also in the microbial world with all the sort of movement of genes. But anyhow, here we get this network where the nodes are the repertoires, and then we have weighted links that tell us how much two repertoires overlap, and we have these bidirectional links, just because we may have some repeated genes. That does not matter so much. What I wanted to say is that, of course, if we start with a medium gene pool in this assembly process, we will see that we get some clusters in these networks. By the way, we take the network And we threshold, I should say, because otherwise everything is connected to everything, we only keep the links of the strongest above a certain threshold to sort of have the interactions that are still the strongest. So the most similar, what's the structure of the most similar parasites? You can see in red a typical, just here, example chosen from a model, where you see some degree of clustering, and then on the blue and yellows, the corresponding neutral modes. Now, a degree distribution or anything would distinguish these networks. But when you get to systems with a very large pool of variation, then the difference between the networks is not so evident. This, I'm just illustrating here with the degree distribution, where of course you cannot distinguish it. If you look hard enough, you may say there are differences in this network. Can we tell them apart? So we built a network classifier based on an ensemble of network properties. For example, we can give them, we can consider three-way motifs in the networks and other properties. And I will not go into details. If you look here at the bottom, in the right, just to mention that we use a number of network features, not just on motifs, but distance, reciprocity, transitivity, a bunch of, as I said, a bunch of features. And here on the left, you can see for high diversity, the scale going from light blue to purple shows you how many times we properly classify a simulation. If we give it blindly to the system, can we basically classify it to the right model for different parameters? And you should just see that we do, and that how much the network features contribute to the classification is here on the bottom right. You see that the motifs are important, but also some other distance-related properties. So we said, okay, we have this classifier. What happens if we apply it to real data? And this is work, of course, in collaboration with Karen Day. We have a long longitudinal study now in its 7 years, so that's nice, here in northern Ghana in the Bongo district. So we built this classifier, and then to be able to classify the empirical system, we used a method called discriminant analysis on principal components. That's what we use for the classification. And if I gave the reference here at the bottom. This method is a very nice method to find groups in genetic data. It was specifically developed for this, and it combines the two multivariate methods, discriminant analysis and principal components. I will not get into details. I would be glad to discuss it. But you can see here in the colors the two principal axes that are from many simulations. The points are different simulations. In yellow and blue, the two neutral models. In red, all the simulations in a certain feasible parameter regime for our system, where you see the difference. And the black dot is the empirical data, which classifies with very high probability to this immune selection regime. So this is telling us the networks, in the network we observe of this similarity between repertoires seems closer to what we expect under those conditions. Now, as I mentioned, there were no clear clusters. It's not a simple cluster structure. And a way to see it is that we are looking at limiting similarity, but in this limit of a very high dimensional space, and it looks, it doesn't look cleanly like clusters. So we did look at the structure. That was the structure in a given season, in a given time. We can look at the structure over time, where each network is built for, let's say, a different season, and we can connect them we can connect them also through similarity, the same similarity, the same threshold as we did before. And what I'm going to do to show you very quickly in this paper is if we look as a function of time and we look at modularity of these networks with a network approach that basically relies on a random walk, we identify clusters and we can, well, modules, so groups of repertoires that are more similar to each other than to members of other groups, and then we track them in time. So these are modules in time with a multi-layer network. And I wanted to show you that although we don't have clear clusters, if we look at the modules, there is a large number of modules on top here of the left. When we have immune selection, we see these modules persist for a while. At some point they go extinct and others are arising in this assembly process. To the right in B, on top, you see these much less persistent modules in the neutral model. So there is a certainty in this very sparse, I would say, in this system where everything is so different from each other, there is something you can call strains, but it's perhaps not what we have been used to think about. So anyhow, let me just pause here and just say that, of course, we don't have the simple clusters, but we have some limiting similarity structure that is affected by this cross immunity. And we seem to have it in nature. So I said this structure is both non-random and non-neutral. We have some sort of clusters that would constitute, it's the best you can find that you could call strains, and it's very dynamic. So, okay, you can say, does this matter at all? And this is largely, now I'm going to move to unpublished work. I like to show you one example that this matters to a response to perturbation. So the kind of perturbation we have here is one that reduces transmission. So if you look here to the right, you see transmission is seasonal in our model and in the real system. And we parameterize this from the real system, including during an intervention that can last a certain number of years, from 2 to 5 years. We use this intervention that reduces, it's known as IRS for indoor residual spraying. We are essentially decreasing the number of vectors, the transmission rate for a while, and then we release it again. I like to show you that in the model, if we compare the response of the system, one of the neutral models, the generalized immunity model, where there is no specific interaction, just a generalized interaction, then what we find is some increased persistence under this negative frequency dependent selection. So increased persistence during the intervention. So if I reduce transmission, which is equivalent here to reducing the growth rate of the parasite, right? For 2, 5, or 10 years in the plots, and I vary, for example, the initial pool size of the system, you can see that the extinction probability in blue for the generalized immunity tends to be higher than for when you have this kind of frequency-dependent selection. So there is this this higher persistence of the parasite through the intervention. And it's not due to abundance, because the prevalence, as you see at the bottom, is very similar between the two systems. So this you can ask, well, why is this? And I did, oh, sorry, perhaps I did, let's see. Well, this is, sorry, I should have added here the reference. This is a paper now in review in Frontiers in Ecology. so you, sorry, I will add that. But what I wanted to show you is that if we look at this quantity I showed you before, which is a measure of overlap, the distribution of overlap, if you look under selection here at the bottom, the distribution of overlap before intervention is in yellow, during intervention in purple, and then after intervention. And you see that there are in the population parasites with very, very little overlap because of this competition. And that is quite robust during the intervention. Of course, it changes afterwards because of the transients, but also because you have lost a lot of diversity during intervention. But this reduced overlap gives you longer duration and therefore higher fitness, which allows you to make it through the intervention. And this is not the case for generalized immunity. You cannot maintain this fraction of the population that is extremely different from each other. So in some way, this structure would, this limiting similarity would enable persistence. Now, I like to show you, and this is the part that I'm most interested in showing you at the moment. We discussed this at the end of Daniel Fisher's lecture yesterday. What about the diversification, the accumulation of innovation in this system? And this is the connection between a force, this frequency-dependent selection is acting at the level of the individuals that are our phenotypes of interest, but it's also acting at the level of the genes, right, that are part of the pool of variation. So I'm going to kind of try to show you that these things are connected. And I'm going to speculate even further and say that a system that is hyperdiverse in nature will always be built from many, many pieces, a lot of genetic variation and phenotypic variation at the lower level, what Daniel yesterday called a nanophenotype, because the same force, you cannot have one without the other. And here is the idea. There will be a threshold. I will show you that there is a threshold. This is in a paper now in review by Shishin and I, and you see that this idea of a threshold in the diversification of the genes, right? So below the threshold, a new gene comes in, but it goes extinct. So these invading genes come in, but cannot stick around a lot. Above the threshold, by the time a new gene goes out, others have come in. So you can accumulate novelty in the system. We wrote this by analogy to, perhaps not a complete analogy to with R0, we wrote it as something we call RDiv. And this is the sort of reproductive number or an innovation number for new genes. You have two components, GNU, and the new rate of gene accumulation, and TNU, the time, the average time that one of these new genes stays around. In this paper, we derived an expression for GNU. I don't have time here to show the details, but it relates on the basis of population genetics on the size of the population, size of the parasite, mu, the rate of change, a mutation rate in the broad sense, here a recombination rate, and then this P invasion, which is the probability that something new invades. And that depends a lot on how many susceptibles you have. So that will be influenced by, again, this kind of selection. And then TNU, the expression for TNU, we got an approximation from some very nonlinear PDE in the supplement, but we don't have a closed form solution for TNU. So computationally, I like to show you that there is a threshold, that threshold, so when this number is smaller than 1, you cannot accumulate diversity. So here it is in the log of our div, so the threshold is at 0. And then what I'm plotting from my simulations in the y-axis is the percentage of new genes during a window of time. So choose a window of time and then count how many new genes have accumulated by the end of this simulation. And I should say that the points here are different simulations with very different both parameters and assumptions. And we see that below this number, there are no new genes accumulating, below 0, and above they do. And what is interesting is that on the right, I have plotted something similar in the y-axis, but for a quantity that measures the intensity of transmission. This is the number of infected bites per unit of time in malaria. And this is interesting because this tells us that as we reduce transmission, we are going to push the system below this threshold. This threshold is well above R0 of 1. So this is a system where transmission occurs, but you have lost the ability to accumulate essentially the building blocks of diversity. And this is illustrated here. In the simulations, each color is a new gene coming into the system. I'm plotting the frequency, and this is over time in a simulation over the transients of this system. The gray color, we are accumulating all the genes that are at very low frequency. The colors are those that have higher frequency. And you can see below threshold, things are coming in, but they are not sticking. To the right, this system is very happily accumulating diversity. So you can ask, does this matter for population dynamics, for what I'm going to call epidemiology? Well, we only have some preliminary results on this. And what you see on top is just over time, a system now with an intervention where at this time 0 here, we are going to reduce transmission in a way that, for example, doesn't cross this threshold in the left or crosses this threshold. So we see before the genes that were there, the frequency of the genes that were there before intervention, and those that now are there building up after intervention. And again, when we cross that threshold here on top in the right, you see that you cannot accumulate new genes. We did here in different colors, you see in the plot with the bars, how much we have reduced this quantity RDIV in these 4 different simulations. Only in the green one, we have crossed this threshold. And I will focus now on the center plot here at the bottom to look at what has happened to prevalence after that intervention. And you see that in all the simulations that have not crossed, we get a reduced prevalence and then the system rebounds. Very characteristic of all the efforts to intervene in these very high transmission regions. You release intervention, the system comes back. Interestingly, in this, when we in the green simulation, that does not happen. So in practice, there is nothing in the system at the population level that is easy to measure that will tell you have crossed this threshold. But it appears to make a difference, and a difference we need to investigate further to how the system responds to these kinds of interventions, and in particular, how fast the system rebounds, or whether it rebounds. So let me note here, of course, that for a while here, these systems, because I should have said, This is not a simulation where we let the system rebound, I'm sorry. We have just reduced transmission to a lower level. And this is why you see that the prevalence basically reaches a different sort of steady state after intervention. We have sort of decreased diversity and therefore, and we have decreased transmission, so we get to lower levels. So let me sort of try to extrapolate some thoughts here that I think apply to this system, but may apply to others. These hyperdiverse systems may well occur at the opposite end of neutrality, where coexistence is assembled under interactions that are specific and frequency dependent. So interactions that will lead to this negative frequency dependent selection, which is a form of balancing selection and therefore should enable diversity. It is in these evolutionary systems along the lines of the stabilizing competition, for example, of Chesson. I think more importantly, large phenotypic diversity in this system is built from a large pool of diversity at the lower level of organization. And this is not by chance. This is not by chance, because I just showed you that the ability of the system to accumulate diversity with this kind of critical threshold is going to also be influenced by these same interactions and selection. So this would set the stage for the existence of an unappreciated threshold that concerns the accumulation of genetic variation on which high biodiversity is built. So that when we lose diversity of species, for example, we may be losing much more, because we may be losing the diversity of the traits that allows the system to have that coexistence in the first place. And the question that is worrisome is, how do we know that we have crossed this threshold and transition to increased fragility of the system? I think that's important.
There is a question in the chat by Aditya. So how do you parameterize mutations? Are mutants related to the parents at all, or are they totally new types?
Yes, thank you very much. That's a very good question. No, they are related to, to the parents, because remember, and of course this, that each type has two elements, so it's very hierarchical, allowed us to do recombination. And so, so there will be some similarity, and in fact, we use an empirical sort of result from recombination on that, that for example, if I'm trying to remember the details, but not all the recombinants are viable, right? Like something very far from the parents is not going to be very viable. So we are kind of, it's not a completely new type. And then, you can decide, you can sort of measure similarity at that level of the epitopes. We have some evidence that in this part of the gene, there are two epitopes, so two parts of the molecule that the system recognizes. There may be more, but we recombine them, and then we have some similarity to the parents as a function of that. We also have mutations, but they are not so important in this system. The details of that mole are described in the paper by He and collaborators in Nature Communications, 2018. So if you like to see more details of this, that's the place to look.
Okay, Aditya say thanks you. Thank you.
Thank you for the question. I forgot. I'm trying to give here the big picture, but yeah, obviously I think some of these results will not be dependent on these specific assumptions, but I think we have to consider them in particular when trying to parameterize the system and so on. So this question of the increased fragility, of course, here at the moment we have only preliminary results on this. I have shown you some preliminary results. We know in malaria that when we intervene, we move the system towards lower diversity of the parasite when we reduce transmission. And we know that the low transmission systems that are, so for example, the low transmission systems in continents where, for example, transmission is lower, like South America, or geographic locations of low transmission, are systems that respond more easily to intervention than these very high transmission systems. So there's an interesting question of could we determine, how could we determine that we have crossed this kind of threshold so that then we could say the system is at a stage where we should hit it with further intervention because it is in a susceptible place. Of course, for conservation, we would like the opposite. Let me now mention that I like to end with some references to other hyperdiverse systems with this kind of selection or interaction, other ecoevolutionary systems like this, for some of you who may be interested. These appear very, very recently. In this work, we look at the strain diversification of a microbe and a virus through the CRISPR-induced immune memory of the microbes. So we know microbes can acquire memory of who infects them, of the viruses, and this is also a very piecewise system, a very combinatorial system, where the microbe basically takes parts of the genomes into its genome, and that constitutes memory. So we work here with bipartite because for microbes and viruses, we can look both at the host and at the viruses. We can look at bipartite networks, and if you are interested in this connection between the ecoevolutionary dynamics and how you see the network structure that arises, we looked at this here. It's a much more dynamic system with different dynamical regimes that alternate until the viruses go extinct. And that may be because this is much more of a predator system, a predator-prey system than the disease systems I was showing you before. Just to end, this is another paper. It's not of mine. But it's a phenomenal paper by the group of in Harvard, Mark Lipsit and colleagues, on another pathogen that is incredibly diverse, Streptococcus pneumonia. They have been looking a lot at how these systems respond to vaccination. You vaccinate with specific strains. How does the system respond? Why is this paper fascinating? And I think it is because it gets to this very important question of these niche differences or this frequency dependence. How high dimensional are these spaces? They look at the genes of these microbes that are non-core genes. So they are believed non-essential. They are not present in all the genomes. They looked at, I forget if it's 3,000 or 4,000, at that point it doesn't matter, and they wrote a system of equations based on the replication equations, which on the basis of frequencies will predict which strains would come out from this vaccination well, empirical system, in the real world. And they managed to do so. So this means that some very effective frequency dependence is being played by genes that we don't even think are so important. It must be through myriad interactions, maybe with viruses, with who knows. I can't begin to kind of think how this is happening. Very, very interesting paper, and I recommend that you look at it. So I like to end here and just recognize, I don't know if I went too fast or too slow, but maybe there is time for questions. This is, I like to acknowledge, as I mentioned, Karen Day and her group for all the very interesting conceptual and empirical work. And thank you for listening.
Thank you very much. So it's open for questions. So maybe I can start. I think you showed this data from Congo, and where there was this very high diversity, and which you could quantify. And could one use data of this type to quantify this? At least maybe not the threshold, but relative. I mean, how populations stand relative to each other.
Yes, that is the case. By the way, I didn't put the slide here. I thought I had put the slide here. If you compare population, of course, if you compare systems, parasite populations in different geographical regions, right? It has been done. I wanted to show you where we got this information on the number of genes in different places. So what Karen does is she sequences the genes, looks at, counts as different genes, those that differ at a certain level of sequence divergence. And then she counts them, and she does the typical cumulative curves that we do for species, but we can do them here for genes. So I wanted to show you those curves, but I didn't put them here. So they are typical cumulative curves, and you can do some extrapolation on how, at what level they should saturate. If you go to South America or New Guinea, those curves saturate at much lower levels than for West Africa. Very, very clear difference. So yeah, biogeographically, we know that. Now, of course, we are saying that that this transition is not just a continuous transition, that there is this threshold that should apply, right? Now, so can you apply that in time to a system in a transient state where you intervene? And now the problem is you could monitor, of course you can monitor, as we are trying to do, the changes in the gene diversity. But in terms of the accumulation of new genes that are coming in, right, you need to do that over a certain amount of time. So it's not a very practical way to find out whether you have crossed this threshold. There must be other ways to determine this, but certainly the point we are trying to make is that we should be monitoring this diversity, which is not typically done because this is such a complex system that it's typically not even considered in epidemiological models of malaria, right? I mean, we at best deal with generalized immunity.
Yeah.
Sorry for a long answer.
Question from the chat. The question is from Pavel Yomi. When we are below the threshold limit, is it possible for the generic system to go to extinction?
Yes, of course it is. And the thing that is interesting, maybe she meant the ones with, when you say generic, perhaps you meant, maybe I'm going to answer that in two ways. For the system.
No, sorry, for the genetic system.
Okay, thank you. So So it is possible, but this is what is interesting, right? And I think this touches on some open questions at the moment, is that threshold. So that when you mention extinction, you are talking about demographic extinction, right? And to some degree, at least from some perspectives, R0 or R would be more important there, right? So that's a demographic number. And of course, you can look at extinction in a stochastic way and so on. That's purely a demographic phenomena, right? Here, this system, as I said, you can cross this threshold at levels where you are transmitting still very well, right? But you are doing so with much less diversity. So it is somehow of a hidden threshold from the population dynamics, but we are saying that it should be important. We have begun to look at that. It's very early work when I showed you that the system that has crossed it essentially stays at the lower level, right? And has a different response to the perturbation. So presumably we know, I'm going to speculate here, the biogeographic regions that at steady state have very different levels of diversity, have over very long evolutionary times, and large regions in some sort of meta-population, right, have assembled much less diversity. So they live in, some of those may exist below this threshold, right? And we believe the ones that in West Africa that are still transmitting at very high level are on the right. I like to mention that some of the pieces of these genes that we find in humans, of the VAR genes, can be found in parasites of primates, so that the phylogeny of these VAR genes can also have very, very deep branches, because this kind of balance in selection will promote persistence, not just of the parasites that coexist, but of the, once you have become part of the established genes, you can exist for a long time. And so that's why also you can accumulate diversity. And I think that, yeah, these are all open questions. Sorry, I took long enough. Are there other questions?
Thanks. So there are no other questions. So you can either raise your hand and open up your microphone, or write it in the chat. So looks like there is no other questions. So so I have a curiosity, Mercedes, if I may. So you spoke about this synchronization phenomenon that occurs inside the body, where this parasite expresses different epitopes at different times. So how does this? How is this synchronization achieved?
Yeah, that's a fascinating question. By the way, there is a body of theory, but I cannot. I don't think there is a full answer. I mean, there isn't an understanding of that, right? There is a literature on that. There are different hypotheses. And the question, of course, one critical question is on which the different moles differ, is whether the immune system is involved, right? So is it something just from the parasite, or is it
a response to the immune system?
Right. And so, of course, it's not completely perfect, but there are waves of parasitemia within the host that are dominated by some of these types, right? So we have, I must say, of course, you notice.
No, it looks like this. I mean, this synchronization inside the host and the diversity, they are a key factor, right?
Yes, yes, of course, the synchronization is very important, because if you showed your 50, I'm going to say 50, if you showed your 50 faces at once, right, then the immune system could react, could protect here and could shorten the infection much faster, right? And in fact, I should say, but of course, this sequential, for the theory, of course, here, because the fitness is related to duration of infection. But I showed you models in which, the other day, in which the memory affected the risk of infection given contact. So the transmissibility and not the duration. It doesn't matter, because the fitness of the parasite is the product of the two. So I strongly believe that the results I just showed you would extrapolate to other systems with different assumptions, as long as you had this competition that depends on overlap and distance.
Okay, thank you very much. So I think we can stop here and maybe go into breakout rooms, and then we reconvene at 6:15 European time for Danny Fisher's second lecture. Thank you all. Thank you, Mercedes.
Thank you. Let me stop sharing. Am I still sharing? No, I'm not.
Matteo, you can see my screen and
yes,
hear me.
I can see your screen now. I'm going to call people from breakout rooms.
Okay.
Oh, maybe there are no breakout rooms.
I don't know. I got invited to one, which I didn't join, but.
Okay.
I was in one too, and then I returned, but I was by myself, so. So something is going on.
Yes, so they've been closed. Yes, people are slowly coming back. I just wait 1 more minute.
Actually, can you see me also when I talk? Because I don't seem to see myself. Is it?
Yeah, we see you. I had the same problem that if I don't leave everybody, like, in the view that has everybody vertically or horizontally, I don't see myself when I'm speaking. So I had to leave everybody, which covers part of the.
Oh, I see. Okay.
I don't know why. Otherwise, you see just Matteo, but we see you.
Okay. Usually, I've been able to, but I don't quite know why. But well, yesterday I wasn't. You couldn't see me talking anyway. So today is I'm trying a different setup, which hopefully will work. So it's.
Yes. Okay, I think probably we can start because I think the number of participants is not going up. So it's a pleasure to have Daniel Fisher for the second lecture on high dimensional ecology and evolution. Thank you, Daniel.
Okay, thank you. So I'm trying a different setup today with my computer, so hopefully it won't cause problems, but let me know if there seems to be any difficulties, and I can try to switch back to how I was doing it yesterday with some time delay. So yesterday we talked about models in which only had one strain, and when a mutant came up, if it was doing better in that environment, then it would replace it. This would change the landscape by a small amount, and that would change the future evolution. And I'm sorry, I didn't actually give this the model a name. This model one could perhaps call a fitness snowscape. A landscape is something which is static. A seascape, which people talk about, is a dynamical environment. But here, it's the evolution itself that changes the environment. So, like when you're going up a mountain and you change the environment that you're walking on as you walk. And what I showed was that the simplest model, and I should emphasize the particular model I wrote down with sort of the cubic interactions, is the simplest model. And that exhibited the red queen phase where the evolution just continued without slowing down. And this, I said that it was very robust. And what I mean by that is if I change the model somewhat, or do a different range of models, then we'll get the same behavior in the limit of infinite dimensions. I raised the possibility that there might be some other behaviors as well, but didn't talk about those. And then I left it open a question about diversification, which I know almost nothing about, but I think is the most interesting question for those kinds of models. I should mention none of this is written up. I think someone asked me for the name of the model, so I guess this is the closest to being the name. Okay, so what I'm going to talk about today and tomorrow is much closer to things people have been, several speakers have talked about so far, which is really talking about a situation where at least I've initially got a fixed environment, but I'm going to have multiple strains. And here I'm going to emphasize calling them strains rather than species, because I'm mostly going to be interested in one species with many strains within that species. Okay. And I'm going to talk initially about assembled communities where there's no evolution. So again, this is what several people have talked about, and introduce a random lot of Volterra models, which then we need to talk about the statistics. And they have a stable phase, as Stefano Alessina talked about, but then that can be unstable. I'm going to then talk about a special model, which is the perfectly antisymmetric interactions. Then more generally, that show that the chaos that occurs in that diverges, and then how this can be stabilized by migration. And that's sort of going to be the main thing I'm going to get to. So I'm going to introduce the sort of phenomenology and the basic behaviors with some rough explanations. And then I'm going to go and talk about the theory and using the dynamical mean field theory. And this is a theory, the method is also something which was applicable to what I talked about yesterday, but there I didn't explain how I got anything. And then I'm going to turn to looking at slowly evolving communities that I'll get to tomorrow, and also considering sort of phenotype models which would determine the interactions. So I should mention, so all of this work that I'm going to talk about today, at least all the completed parts, was done by Michael Pierce and Natish Agarwala, and is published in PNAS earlier this year. Okay, so the models that we're going to talk about, the generalized Lotka-Volterra models, and I'm going to call the number of strains K. So I labels those, runs from 1 to K, and Ni is the population of strain I. Now I should apologize, is this different notation than Stefano used. He called the populations Xi and called the number of strains N. Unfortunately, there's not completely standard notation. But I'm going to start down writing the model as he did. So one has the basic growth rates, or growth and death minus death rates here, Ri, and then the interactions between the strains. Okay, but we're going to be interested in the closely related strains. Okay, so if I have closely related strains, then it's natural to replace this by things which tell me how the strains are similar to each other. So I'm going to write that Ri then, say, is going to be equal to some R bar, which sort of average over all the strains, that's some common part, plus some varying part, plus some part which is varying, which I'm going to call Si. Okay, and I can think of the Si then as being like selective differences between them. And the natural one is then to take the Si with an average of 0. So that's going to be the variabilities, the way the strains differ from each other. Okay. So that's going to be my ARs. Now, what about the interactions? Okay, well, the interactions, I'm going to consider two parts. So I'm going to take the Aij is again going to have some overall average interaction, which will generally be negative if they're competing with each other. That'll be less than 0. And then they'll have a strain-specific one, and I'm going to put a factor of n in here just so I don't have to carry factors around n anywhere else. And this is going to be the interaction with itself. So Q is really going to be the sort of niche interaction, or 1 over the carrying capacity of that strain. Okay, and then we're going to put in some random parts, and again I'm going to pull out a factor of n out in here. So the Vij's are the between strain interactions, and so the Vij, again, I'm going to take to have mean 0, and then I'm going to say something about what the statistics of those are. Okay, so the Vij's are going to mean 0, typical magnitude 1, sort of sets the overall scale. Now, since these are going to be considering different strains, these are going to be much larger than the differences. So this and this is going to be small compared to these. And so what does that mean? That means that the R bar and the A bar basically set the total population. They constrain the total population because they're the bits which are the main interactions with the growth and the competition. So they're going to constrain the total population, and that I'm just calling this big N, which is the sum on I of the Ni. Okay, and then I'm going to consider what they essentially do is make this approximately constant, and then there are variations, of course, between the Ni, but with the N being roughly constant. Okay, so in that case, then it's natural to define the frequencies. So I'm going to define the frequencies, which are the fractions in the population. So I'm going to define nu I as being equal to Ni over the big N. So it's the fraction of the population. So obviously the sum on I of the nu I equals 1. Okay, so that's going to be my my model, and then I can rewrite the model in the slightly different form. I'm combining these. I'm now going to pull out the R bar and the A bar, and I'm going to write the model this way: is the di ni dt, and I'm also going to use the notation of a dot for that. So that's going to be nu I. Okay, times. Well, now I've only got the differences coming in here. So I've got the Si, and then I've got the plus the sum on the j of the Vij. Oops, let me put them in another order. This has the minus the specific one, the q times the nu I, and then it's got the plus the sum on the j, Vij, nu I, nu j. Okay, but then I'm going to put an extra piece here. Okay, where epsilon is a Lagrange multiplier, and the role of that is to make n equals constant, or more specifically, enforce this constraint that the sum on the nu I is 1. Okay, so I'm replacing the effects of the overall growth and interactions, and I'm only going to consider the effects of the differences. So this is the differences in the growth and decay rates. This is the interaction with it themselves, and then this is the interaction between the types. So this is the model which I want to understand, and this is going to have average 0. This is going to average 0. So of course, what we have to talk about is we have to talk about the statistics of the interactions.
Daniel.
Yes.
Then so. Do you really have Vij, nu I, nu j there, or is just nu j? Because that
So,
would be a
thank you. This is the drawback of writing things in real time, is one keeps the audience on their toes. So thank you for correcting that. Yes. So that's just a standard, the standard interaction. And so the first term, this term here would be, you often written as like effectively like 1 over the carrying capacity, but I've rescaled things, real scale things out. So I don't want to keep things all over the place. Oh, so I should say one other thing before talking about the statistics. I'm assuming no stochasticity at this point. Okay, but I will want to consider extinctions if nu I becomes less than 1, i.e. less than 1 over n, which is corresponding to ni less than 1, of course, then I can't have less than 1 individual that'll go extinct. So I'm going to mostly consider the deterministic interaction, but then I'll talk about the effects of the extinctions, which of course can play an important role, particularly because they'll happen even in these simple the simple models. So this is my basic model, and the thing which I then have to say some things about is what the statistics are. I've said the interactions are going to be interactions, and the SIs are going to be random. Okay, so the SIs are going to be independent. Okay, and I can talk about the variance of those. The variance of SI is just sigma S squared. Okay, and these I would expect that they'll be small. Very small, because the strains are all similar. Okay, so this is sort of a consequence of the prior evolution, that they're all going to be very similar to each other. If one was better, that strain would diversify and so on. So this is going to be a basic assumption that's small, and to often I'm going to set that equal to 0. Okay? But the crucial bits are the Vij and what the statistics of those are. Okay, so again, I'm going to take for I not equal to j, so that's the interactions. I'm going to take Vij squared equal to 1. So the variance of the V's is just equal to 1. So this just sets the overall scales of the time. Okay, but the crucial bit is then what are the correlations? Okay, so in particular, I want to ask what is the correlations between Vij and Vji. Okay, so this is going to be the only correlation which I'm going to put in. So this is going to be the only correlation. It's going to be between those. So this is how the correlation between the effect of strain I on strain J and the effect of J on strain I. Okay, so if this is going to be competitive, then I would expect this to be positive. Okay, so I'm going to define a parameter gamma here, which is going to be that correlation. The competitive would correspond to gamma being bigger than 0. If I correspond to gamma equals 1, is totally symmetric. That's correspond to a symmetric Vij, and that's often what's considered for models with competition. Okay? In general, gamma lies between -1 and 1. Okay.
Sorry to interrupt you.
Yes.
Shouldn't Bij also be small, so the variance of Bij be order 1 over the number of species, or?
Well, that's a choice of a convention. That's a convention which often people choose for being useful. I want to talk about more things in terms of unrescaled quantities. So the interaction is setting the basic scale. There's nothing intrinsically that says it's going to be small. Okay. Now, since the new i's, if there's many species here, the new i's are going to be each of order 1 over k. Then this interaction piece is going to be small coming from that with these random of order 1. So this whole piece is going to be of order 1 over square root of k. But I explicitly don't want to scale out the k's. It's convenient for doing theoretical analysis, but I don't want to do it because it sort of confuses what one's assuming when one does that. Okay. Of course, I can have k, 2, 3, 20. I can add more things. I don't want to be rescaling the v's each time I add more things or take more things out. So these are, I'm trying to keep it in terms of the physical or the biological quantities. Okay, so gamma equals 1 would correspond to symmetric. What about gamma is less than 0? What would that correspond to? Okay, so gamma less than 0. So this has 2 possible things it can correspond to. One of them, for example, is if I have a direct competition. So if 1 beats 2, okay, what that means is that V1, 2 is bigger than 0. But of course, that means 2 loses to 1. So 2V21 is less than 0. Okay, so that's a natural way in which you can get anti-symmetric correlations in the matrix. Okay, I just want to add an extra page in here before I have my next bit. Okay, so that's one possibility. But there's another possibility, and this I'm going to talk much more about tomorrow, and this is what makes it much more interesting. There's another possibility, which is I have bacteria. So I have bacteria, and so those bacteria will have populations, say Bi, and they'll say B K of the bacteria, a number of those. And then I have phage with populations P sub L, and I've got some number of those. So I've got the bacteria and the phage, and these interact with each other. And so if I look at the dynamics of those, then Bi dot would be Bi, okay, times some growth rate for the bacteria. But then there'll be minus a term which is coming from the interaction with the phages. So this would be some matrix here, HIL times the population of the phage. Okay, so that would be that term, where these are going to be positive because they're negative effects on the bacteria. And then I've got the phage. The PL is going to be, sorry, I also want to put a term in here, which is a stabilizing term, sum on J of NJ. So that's coming from the competition, BJ, okay. Okay. And then I've got the phage. So the phage are going to die at some rate. The phage are going to die at some rate, but then they gain by eating the bacteria. So they have a sum on I here of some other matrix. Some other matrix F. Now I L I times the population of the bacteria. Okay, and again, these are going to be positive the way around I've got it. Okay. So if I think of that as putting all these together, and I'm going to think of these as being strains of one species of bacteria and one species of phage, so I now have a two species model, but with diversity within the two of them. And if I look at what the interaction matrix is going to be there, so what is the interaction matrix going to be in that form? Okay, well, it's going to have this form here. If I think of putting the bacteria on top and the phage underneath, it's going to have some minus constant all the way through this part from the bacteria interacting with each other. Then it's going to have minus the H from the bacteria interacting with the phage. Over here, it's going to have the F from the phage interacting with the bacteria, and there are no specific interactions of phage with these other. Okay. Now, what do I expect here? I expect that F will be approximately F bar plus something small, so delta F I L, and H will be some average value plus some small variations. So again, in the same spirit. Sorry, I've got my my indices backwards. So I have these here, and I would expect these will be correlated. So more specifically, F and H transpose are correlated. Okay, that's corresponding to the fact that, of course, if one phage does better against the bacteria, that bacteria is worse for the phage. Okay, so if I look at this whole matrix here, it has anti-symmetric correlations in the dominant parts here. There's also this part which is symmetric, but there's a dominant effect here, which is the anti-symmetric ones. Okay, now it turns out this model behaves very similar to the simple random Lotka-Volterra model that I'm going to mainly talk about, and I'll come back to this one some tomorrow. So you can think of this as being one of the primary motivations for considering models in which gamma can be negative. Okay, so one thing we can then start to talk about is, what does this model look like? So I'm going to focus on this model here. So we've got here we've got two main parameters. We've got this parameter gamma here, which we can make in red to define that. I've got that parameter is the key parameter. This upsilon is just the Lagrange multiplier that adjusts itself. So the only other parameter we've got here is this Q, which is this niche interactions, and that's our basic parameters. Okay, so we've got a model now with two parameters. One is the interaction within the same strain, between same strain, and one is the interaction with others. Okay, and I generally have then the S's as well. So I have another parameter which is associated with the S's, but I'm going to mostly consider the simple possibility where the SIs are 0. There's no overall differences. We can add back in the effects of those. Okay. So now I'm just going to show a phase diagram, and I'm going to say how one gets it in a while. So the S's are equal to 0. So we've got our two parameters here. We've got the Q, which is the self interactions. That's the strength of the niche interactions, and then we've got gamma, which is the symmetry. Okay. Well, if the interactions are very strong relative to the within the strain, very strong relative to the interactions between the strains, then we've got the standard behavior and the limit of a large number of species. We've got a, so this is now we're going to want to consider K, the number of species, being much larger than 1, the number of strains much larger than 1. And up here then, in this regime up here, we've got a single stable community. It's a large community, stable community. And that's actually with high probability for the limit of large numbers of strains, there's going to be a unique community there. Okay. So this is a result which has been worked out by many people, particularly a lot on recent years, but is more or less something which May already Robert May already knew. Okay. But then there's a boundary, which is where that goes unstable. So there's a line I can draw across here. And this is a line here. And this line goes from 0 to a value over here of Q of square root of 2 times K. And so here's where the square root of K that Matteo was asking about comes in. So this is when Q is bigger than this, I have a single stable community. But when I lower the Q and I cross through here, then the system goes unstable. I lose the single unstable community, okay? And then the big question is what happens down here? Okay, what happens under there? There's not a single large stable community. That's the one thing we know. Okay. Now there's a special line. There's a special line which is corresponding to being along here. Okay, that's the line where it's the perfectly symmetric model. That's been studied by a lot of physicists also recently. And in that case, what's known, so this is for gamma equals 1. For gamma equals 1 here, there are a very large number of stable communities. There's actually exponentially many different communities. Okay, but that's very special, and it turns out that as soon as you go away from gamma equals 1, things change. This behavior over here has a lot of similarities as far as the dynamics to the things I talked about last time of the random landscape. Is you never actually get to one of these communities. You sort of wander around, things go bouncing down, things come back up again, and it never really settles into them. But if you just look in terms of the communities that exist, there are exponentially many stable, uninvadable communities of subsets. Okay, so it turns out this this community up here is going to have, in this case here, it's the number of the size of the community. So the number of which persists in the community is going to be bigger, greater than or equal to k over 2. So more than half the types persist. Okay. As you go down through here, let's say it goes unstable, and we want to talk about down here. Okay. If we ask about putting a little bit of the s's in, so if the sigma of the s squared is not 0, if the sigma s, so the variance of the s's, as long as that's less than or of order, actually it turns out to be order 1 over root k, okay, then you only get quantitative changes to the phase diagram. But it stays qualitatively the same. Okay, but I said we're mostly going to say the SI is equal to 0, and I can talk more about that. Okay. So one thing to note is that you need to have the selective differences being very small to get this behavior. If the selective differences start becoming substantial, then if we go back to our basic model here, that means this term will then tend to dominate, and this term will be small compared to that, because this is sum of a whole bunch of random things. This will dominate, and I won't get the interesting behavior. Okay. Now, what about the Q? Well, here you notice that in order for the Q to be big here, it has to be bigger than a value which is of order square root of k. Okay. So to get this, you need to get the stable community. To get that, you need to have k greater than or equal to of order root k. Okay. So this corresponds to saying the interactions with your siblings, right? These are all different strains. Interacting with siblings have to be much, much bigger than the interactions with all your second and third cousins. So this is basically equivalent to assuming that there are niches. You assume that each one interacts with its own strain much more than it interacts with others. Okay, and there's no a priori reason to assume that. Okay, so the particular things that we're going to focus on here, the bit I'm going to want to focus on is actually along here, along this line there, where either the Q is very small, so we might as well set it to 0. The natural assumption then is if I have these closely related strains, so with the close relatives, the natural thing is to say that Q is very small. Q equals 0. I've just got the random parts with that as well. If Q is of order 1, that doesn't matter. That doesn't matter much as long as it's small compared to root k. Okay, so I'm going to consider that, and then for simplicity, I'm also going to consider the SI is equal to 0. But I say that will come back and re-examine.
So I think you want to get the questions as they arrive. There is a question in the chat. So you can read
yes.
It. It says, as antisymmetry in A still doesn't capture the fact that the signs in the upper and lower triangles for the bacteria phase system are positive and negative. Doesn't that make a difference in behavior?
So the interactions, by definition, the phages are bad for the bacteria. That's their lifestyle. So these terms here, this has a negative sign, and the H's are all positive. Okay. Over here, the phages are eating the bacteria, and so this is those terms are also all positive. What the random parts is in the difference. So this is positive, but then the random parts are associated with differences between them. Okay, so I'm going to come back and talk more about this one this tomorrow. Okay, the other questions on the model or the sort of basic setup. And I'm going to explain a bit of how one gets this phase diagram, but I mainly want to talk about this, focus this part here.
There is another question. Is there any variability in the self-interaction, or is it always exactly Q?
Okay, sorry, I didn't see that one. So the self-interaction there, I can have some variability in it. In particular, if I look at my interaction here, I've got a part where I equals to J, so that would be the variability in it. It turns out if it's variable by similar amount to how the interactions between strains vary, then it doesn't matter much in the limit of large K. It only matters in the limit of large K if it's big, in particular, if it has to be of order root K. It has to be much bigger. So I can turn the other way around, and I can say, let's have fixed Q. That's going to be a property of the biology, a fixed distribution of the S's, and ask what happens if I add strains. So I'm assembling a community here. I'm adding more strains, and then what will happen is I add more strains, it'll go unstable. Okay, and where it goes unstable will be associated with how big the Q is. Okay, so that's that basic results of May. He didn't really quite do things right, but the overall result is he certainly got right. So that's this part of the phase diagram up here, where there's a single large stable community. And what happens down here for large K really only started to being investigated in recent years. And I'm going to show some simulations of that, but then really try to develop a theory. Okay. So we're going to consider the behavior along here. Okay. Okay, so we're going to consider the close relatives. We'll focus on this, and for reasons that sort of motivation of the bacteriophage, we're mostly going to concentrate on this region, here. So we're going to concentrate on gamma being less than 0, and it's between 0 and -1. But actually, we sort of believe that most of the behavior actually persists in this whole region, except on that special line. So I'm going to not talk about the stable community. I'll just say something about how one gets it, but I'm really want to talk about what goes on in here. Okay. So for doing something general, I want to do something very special. So one of the things that one has learned from experience in statistical physics is there's a lot to be gained from having particular models that one can really analyze and understand in detail. And then one can sort of use ways of thinking about what's more general and what isn't to ask which features of those might persist and which ones don't. Okay, so I'm going to first talk about a very special model, and this has been looked at by over many years, a special model, and that's the case where I'm going to have the Q equals 0, but I'm now going to have gamma equals -1. Okay, so this is going to be the special thing. It's perfectly antisymmetric. Now I've got the antisymmetric V matrix. Okay, perfectly antisymmetric. Okay, so this has a very special property. It has various things which are known about it. It has a unique. It always has a unique stable community, and by stable here I mean an uninvadable. Community, and this Stefano talked about in some generalizations of this, and the size of this community is going to be approximately K/2 plus or minus some order square root of K, which will depend on the particular realization. So this community always says it's unique, stable, uninvadable community. Okay, and in that community, each of the nu i's will be some fixed point value, nu I star. So this is then the fixed point of the dynamics. Okay, or nu I will be equal to 0 for the extinct ones, for ones that go extinct. Okay, so if you look at the surviving types, then it's very special. And you can see that this is very special because this fixed point here, this fixed point is marginally stable because of the antisymmetry. It means that all the eigenvalues for the stability around that fixed point are imaginary. So the imaginary eigenvalues, it's not stable or unstable. It's very special. Okay, and that very special behavior is associated with a conservation law, a conserved quantity. And so this is just a mathematical artifact, a conservation law, but which Stefano had talked about. And this this quantity here, which plays the role of an energy. Okay, it plays the role of an energy. So I'm going to call it that, and it's just going to be minus someone I of the nu I stars. Okay, times the log of the nu I. Okay, now you can add another piece to this, which Stefano added. You don't have to include that in this case because the total number is 1. So you've got this population here, and this is conserved. It's a Lyapunov function, but it's conserved conservation law after the extinctions. So after all the ones have gone extinct. Before that, it's a Lyapunov function. It'll increase, but when they go extinct, then it'll saturate and then become a conservation law. Okay, this is the conservation law, and you notice this is a sum of a whole bunch of independent terms. Okay. The other thing to note is that in the log variables, so if I define Li, which is the logarithm of the nu I, So this is just the log variables. They're the natural ones to think about. If I think about populations growing and growing and shrinking at some rate, depending on whether their net growth rate is positive or negative. So this is the LIs, the natural variables. And in these variables, the phase space volume is conserved. Its volume is conserved. Okay, again, it's just a mathematical nicety, but enables one to do certain things. Okay, so this means that there is a steady state of this, which is like equilibrium stat mech of a bunch of interacting of independent particles. Okay, so you can immediately write down the dynamics. There's a quantity which is like the temperature. There's a quantity here which is like the temperature. Okay.
Sorry, Daniel, can
yes.
I ask a question? So
yes,
so the nu I in your equation for the E is a dynamical variable, right?
The nu I is the dynamical variables, right? So this is generally a function of time.
Is
instead the fixed point.
So the dynamics does hover around a fixed point, or does it go to a fixed point?
Okay. So the dynamics, if I look at the simple cases, okay, so I can look at the very simple, a very simple case here, and I can ask what this looks like. And the simplest situation I can got if three of them survive. Okay, let's go down here. So if I have three surviving, okay, three of them in the community, in the stable community. Okay, the sum of those is always equal to 1, so I can draw the flows here in the news in a phase space where this would be pure 1, this would be pure 2, and this would be pure 3, and somewhere in there's a fixed point. Okay, so that's a fixed point, but it's marginally stable, and what that means is there's a family of stable orbits. So there's that one there, or there's another one which is here. So these are all limit cycles. Okay, these are all limit cycles, a whole family of those. So there's a whole family of the states, and that's where it's very very special. Okay, and in this simple community, they're just periodic like this, and this is in fact like the classic Lotka-Volterra model of predator and prey, which has a family of cycles here. So this is like approximately like the original Lotka-Volterra predator-prey model, with one of each, and its dynamics also looks like this, and there's a whole family of cycles. But we sort of know that thing is very special. Okay, so what happens more generally? So there's going to be something which is going to parameterize this this family, and there's something which is going to parameterize this family, which is roughly speaking how big a range they go over. Okay. So this quantity E, it can be anything. It's going to depend on the initial conditions. Okay, so this quantity E here can be anything. Well, it's constrained as to what values it can take, but the V is variable, and so just like in thermodynamics, this is going to classify the what state one is in. There's a whole family of states, right? So there's a family of states. A family of states here. And they parameterize something which is like the temperature, which is proportional to the average of the energy over all of the species. Okay, so the quantity is like the temperature. It'll determine how big these fluctuations are. I'm going to show some pictures in a minute. Okay, this temperature is going to be conserved. I've got simple statistical mechanics. So if I look at the probability distribution of all of these. The probability distribution then is going to be the probability density of all the nu i's. Okay, it's going to be proportional to nu I to the -1 plus a quantity which is going to be related actually to the nu I star over theta, and that's because of the nu I star appearing here. I exponentiate this, I get nu I to a power. And then there's going to be a factor which is just going to be associated with a constraint, which is the delta function that all the sum 1 j of the nu j is equal to 1. And of course, I need a product over all of the I here. So they're all independent of each other. They're independent of each other and this distribution which looks like this. Okay. Rather amazingly, this is identical to the distribution from purely neutral theory. So this distribution is the same distribution you would get out if you just had a large number of species with a bit of migration into them, and they would have a distribution here, which would be a distribution which would depend on migration rate in that case, but it would have exactly the same distribution. They would be independent. So this looks like a neutral distribution. Okay, but this is completely, if you like, coincidental. This is just coincidence because the dynamics here is nothing like neutral. There's no stochasticity. It's driven deterministic dynamics. Okay. So let me just, there's a couple of questions here. So one of them is why is E of the sum of the terms with no interaction? So that I refer you to Stefano's Alcina's talks, where he showed explicitly that this quantity is conserved. It does depend on the interactions in the sense that the nu I star, right? These nu I star are given by the V's. So the Vij's determine those nu I stars. So it does depend on the interactions, but the only way it depends on interactions is those. And this particular quantity is non-interacting. Nevertheless, the species are interacting with each other. The strains are interacting with each other. And so there is dynamics, and that's what we're going to be interested in. Okay. So this is all mathematical. This is just going to motivate some things. If you don't pick up this, it doesn't matter for what comes later.
Yeah, there's another question. Whether it is a coincidence that the formula for E looks like an entropy.
It looks like an entropy. It's not. There's a way of inferring it that it's. You think of it like an entropy, but it's not very useful. I don't find. It's more like an energy. Yeah. Okay. Let's see. Was there another one there? Yeah. So this is like these are the neutral cycles in the Volterra model. They occur when 3 is in the community. Okay. But when you get more than 3, here, so for k greater than 3, well, okay, much greater than 1 at least. So for k much greater than 1, what happens then is you might think that you would get a whole bunch of cycles, different cycles. Remember, everything is interacting with itself. Interaction between cycles tends to be unstable. It tends to drive chaos. And so here one gets a chaotic state, and there's a whole family of them, I say, parameterized by this quantity theta. Okay. So now I'm just going to show this, what this looks like, and then we're going to try to explain it, because this only tells us a steady state. This doesn't tell us anything about the dynamics. It tells us about static snapshots. The static snapshots are not very useful because those static snapshots can easily let us think that we're in a completely neutral model. Okay. However, these are large populations. These are microbial populations. The n's are large. The demographic stochasticity effects are small. So the neutral theory just can't be right quantitatively. So this is a coincidence, but it's a real warning. The other thing you note here is that if theta is very large, when there's a lot of chaos, so theta is very large, then this is roughly uniformly spread out on a log scale. So it's roughly uniform on a log scale. Okay. So that, in fact, one sees in the picture. So here's some numerics for this perfectly antisymmetric model. And here, the characteristic scale of the temperature is basically how wide the fluctuations are on a log scale. So this scale is plotting on a linear scale, and you can see here on the linear scale, these come up, they spend most of the time down here and occasionally burst up. So these all have bursts upwards, and I'm going to say why in a minute. They come up and come back down again, some other one comes up and so on, and you get this dynamics. To really see what goes on, you need it on the log scale. So this is the frequency on a log scale here. So this was this quantity which I defined as being the Li, which was log of the nu I. So that's looking at that frequency, and each of these colors then a different type, and this is a function of time. Okay. So this is the log you see the spread here, and this basically scale here. This scale here is this quantity theta, which is like the temperature. If the temperature is small, they will be at a fixed point. So theta equals 0 corresponds to a fixed point. Will correspond to the fixed point with no fluctuations. Okay, and large theta will correspond to these wild fluctuations. If you make theta even bigger, you get even bigger fluctuations, but they're stable fluctuations at stable chaos. Now, this picture is only after a lot of others have gone extinct. So if I started with others here, some of the other ones will go extinct, and they go extinct quickly and stay extinct. Okay, so in addition to this, I've got approximately half of them are extinct, which I'm not showing. Okay, and this chaos you see by the time you get to relatively modest number of types. And then you get this chaos behavior, and certainly in the limit of a large number. Okay. So what is this this coming from? Well, this dynamics is coming from this anti-symmetric nature of the V. Okay, and this dynamics one can call ecological kill the winner, and is very related to things that Mercedes talked about today, which is that it's the disadvantage of being popular, disadvantage of being in big numbers. Okay, so let's consider one particular strain here in black. Okay, now at this point here, there's a whole bunch of blue strains, which are the large ones. And if it just happens that the signs of V is such that those blue ones favor the black ones, then they tend to drive the population of the black ones to come up. So these blue ones drive the population of the black ones to come up. So this then comes up. But then what happens when this comes up? Then the strains which prey on it, which don't like it, are the red ones here. So then those red ones will come up, because this one is now high. So these red ones will now come up. These red ones will now come up. And what will those red ones do? They'll inhibit the effects of the of the black ones, and the black ones will come back down again. Okay. Earlier on, when the blue one, then the black one first came up, the blue one started to go down. And again, this is because the anti-symmetric sign of the anti-symmetric sign of the interactions. So this is a killer winner. It's something which is there clearly in bacteriophage models. It turns out you automatically get it when you've got these anti-symmetric models. Okay, and this behavior for this purely anti-symmetric model is going to be a clue to behavior more generally. Okay, so the reason I'm going to talk about this is this kind of behavior with these fluctuations approximately uniform on the log scale, as you can see more here, spread out of some range on the log scale. That's going to be the ubiquitous behavior. The behavior is complicated. If you look in the details here, you see all these wiggles. Here you just see what looks like bursts. Okay, and a crucial part of this is that each type here, each strain that survives has a burst up to a bloom up to high numbers. Okay, so if you went on for long enough time, you would see each of these strains coming up at some time. Some of them come up more often, some of them come up less often. Okay, so that's something which is sort of natural maybe natural in the bacteriophage context. Okay, so this is kill the winner dynamics. Why is it called kill the winner? Well, whichever one is high at that time. Oops, sorry. Whichever one is high at a given time, okay, the ones that do well against that, like this blue, red, black one here, those are the ones that will come up. And then because the anti-symmetric interactions, then they'll kill that one. That'll come back down again. Okay, so that's where the kill the winner terms come from. This is used in several, both ecological and evolutionary context, and so maybe it is not the best term to use, but the basic dynamics here is coming from this anti-symmetric behavior. It pays for phages to attack or to evolve to attack the most common bacteria. They do best, and that drives it back down again. And it pays the bacteria to be resistant to the most common phages. Right, so that's the opposite side of it, and that's what gives rise to this dynamics. Okay, but now we have a problem. The problem is this gamma is as soon as gamma is bigger than -1, the behavior is different. The gamma equals 1 is very special. We have no conserved quantity, no conservation anymore. And what happens is we get a behavior like this. That if we look at the log of the new i's, and we look at them here, okay, so that's the maximum they can go up to. So they'll be fluctuating around. So here's one of them fluctuating around like that. Okay, bigger and bigger fluctuations. If I do another one will fluctuate around also, have even bigger and bigger fluctuations. And you get divergent fluctuations that go to extinction. Okay, so you get divergent fluctuations which drive extinctions. Now, this one can very easily see already in with the 3 types. So if we looked when we just had 3 types, 3 types here, you can already see this. So if we've got 3 types which are surviving there, and I look again at the dynamics. So this is pure 1, that's pure 2, this is pure 3. Okay, and in this case, we can have a fixed point in the middle, but that fixed point's unstable. That fixed point's unstable, and if I look at dynamics, the dynamics gets closer and closer to extinctions. So the dynamics goes around, gets closer and closer to extinctions. So this is unstable dynamics here, heteroclinic dynamics. Here it's unstable chaos. It's divergent chaos. There's no steady state. It just drives extinctions. The dynamics get slower and slower, bigger and bigger fluctuations. The deterministic approximation breaks down. And of course, at some point here, I get extinctions if I go below this. Okay, so if I go through there. Okay. So then what will happen is you end up with a few types left. They'll typically have a cycle with a few types in that. Okay. So this is behavior that tells us it was very special. It's pretty useless. So why do I care? Okay. So at this point, we have to ask, how does one stabilize the dynamics? I need to add some. Add a page in here. So how can we now stabilize the dynamics? Okay. So one way to stabilize this, a common thing to do, okay, is by migration. Okay. So the normal thing one would think of is I've got some island that I'm looking at, so that we're looking at, and then we've got some big mainland over here, and I have the species coming in from the migrating in from the mainland, and so species I say comes in at rate mi, comes into there, and this is the island I'm focused on. So we lose the we lose the possible extinctions here because I always get the extinction. So this is a big pool with all of the types, the strains in it. Okay, so what is that corresponding? I'm just adding a little bit of migration term. However, this is cheating. I consider this completely cheating. It's cheating because the problem of diversity on the island is just replaced by understanding why there's so much diversity on the mainland. Okay, of course, if there's geographical structure and so on, that can happen, but we want to understand sort of which things happen in principle and in simple models. Okay, so this is really cheating. So we don't want to do that. Okay. However, we can still think about migration. I'm now going to have many islands. So I'm now going to have I islands, okay, which are labeled by alpha is equal 1 up to I. So those number of islands, okay. And I'm now going to have migration between all of the islands and all the other islands. So I'm going to have migration going in both directions here, migration going in all directions from each island to every other island, so on. Okay, so all-to-all migration. Okay, but things are going to be simple is the interactions are only going to be on the islands. So the interactions are only on the island. On the island. Okay, and all these islands are identical, so I'm not allowing myself to have different environments. Okay. Now, it turned out already 2 islands is interesting, but we're going to consider the case where this is going to be very, very large, mostly. They migrate from all to all of them. And so what does the dynamics look like? Well, if I look at the dynamics for a population on one island, so I've got now new I alpha. So that's the ith type on the alphth island. So that's just going to be new I alpha. Okay, so it's going to have the terms that it's got before. I'm going to ignore the S and the S and the Q here, and just write down the interaction term. So this just has the sum on the J, okay, and it's got the Vij, which is the same on all of the islands, but then it's only the ones on the same island that it's interacting with. Okay, then it's got the a Lagrange multiplier, which is just going to be for that island, which depends on time. That keeps the total population of that island fixed. Okay, but then it's got another part. It's got a part which is m, a migration rate here, and I'm going to normalize it this way, so it's sum on all the islands of the sum on all the other islands of the same species on the other islands. Okay, and then it's going to have a migration out, nu I alpha. Sorry, and this bit doesn't come. This bit comes, of course, outside here. This is not a growth rate. This is migration in and migration out. Okay, so that's the migration effects. They come all to all, and this quantity here. Okay, this quantity here. This is the average over all the other islands, and I'm going to call that quantity nu I bar. Okay, so that's the average over all the other islands. Of course, that can itself depend on time. So that's the island average. That's saying I get input from all of the other islands, and I get migration out. Okay, so now we have to ask what happens. Okay, we have to ask what happens here. So we now model some number of islands. You can simulate the different islands. They're all the same on each, and we can ask what goes on. Okay, so here's this. Sorry, this was meant to be down here. Added the page in the wrong place. So here is now I'm looking at a situation with 10 islands. So we've got 10 islands here. We see an example here where we've got global extinction. This is showing the one type, so one strain across many islands. So this is dynamics of one strain I'm looking at here. And the different colors are now the different islands. And you notice it's bouncing up and down on the different islands. It starts going down. If I look at the island average, so this is the quantity which is the island average, coming from the total migration rate into each island. Sorry, I called the news N here. I didn't have the normalization. And if that fluctuates around, if that fluctuates down, they don't get much migration anymore. And then if these die down, they just go extinct. So here's the extinction threshold when the frequency on an island reaches of that strain reaches 1/n. So this is going to global extinctions. So I haven't made the assumption of the mainland. In this case, in this situation that I've got here, this particular one we've shown is if I look at the island average for this type, this island average goes to 0 at long times. Okay, so this is just going extinction. So I've got a global extinction. Okay. So that's boring. But you can also have the more interesting phenomena. Here's now looking at another strain in the same population, the same community of the strains. So we're looking at another strain here, and this strain, you notice it comes up and down. You can get local extinctions. Here's something which has dropped all the way down to extinction down there. In fact, you can actually get the total population to go down small enough that it would go extinct, but it actually doesn't because it happened to be some strains that are doing well at that time. They come up, they repopulate the other islands, and everything goes along and stays. So this looks as if the dynamics is stabilized. A crucial part here is that the chaos on different islands is desynchronized. Across the islands. Okay. Now it's generally true if you take two chaotic systems and you put a weak coupling between them. So we're particularly going to be interested in the cases where the migration is very small. We're going to be interested in the small migration. Sorry, I messed up the pages. So we're interested in M being very small, less than basic growth rates times. So when M is very small, we get this desynchronization. It doesn't have to be that small to get that, and we can get this behavior. Okay. Now, not only that, but you can actually have a new type. So here's a new type. It's initially coming in on one island. It comes in there, rises up enough that it starts seeding other islands. It actually comes back down again. It goes extinct on its island, but meanwhile it's seeded some other islands. It rises there. Some of those go extinct. But eventually you get to something which is a steady state that looks like this. Okay. So what we've got here is one finds from the simulations, at least, there's a possibility for stable chaos here, We can get stable chaos by desynchronizing all the islands, and we can provide a pool. Okay. So our theoretical challenge is to try to understand this this behavior. Okay, so that's going to be our challenge. That's going to be what we want to try to do. Okay. Now I should be honest here. Being an old-fashioned theorist, I tend to think that one of the roles of theory is to confirm simulations rather than the other way around. If I give you a calculation in detail, you can check whether it's right. If I give you a simulation, it's much harder to check whether it's right. Okay, and in fact, in this case, the theory actually came before the simulations, and then there was a very nice back and forth between the theory and the simulations of some of the things that I'll talk about tomorrow. Okay, so we really want to try to develop theory for understanding this. We want to understand the simpler situation, which is the chaos on the one island in the perfectly antisymmetric case. So we wanted to understand this, and then we want to build on that to understand the dynamics of the models with the on the many islands. And I should be able to move. Well, never mind. Yes. So we want to be able to understand all this. Okay. So this is the basic phenomenology. If I look at any given species, it can spend a lot of time down here, down at the sort of floor that's set by the migration in, as long as it's coming in here. This is the migration coming in sort of black rate, which will set the sort of floor here. And so this floor coming from the migration, they won't tend to drop below that. Okay. So this thing, this dashed line, we can sort of call the migration floor. Floor, that's the lowest it's going to go between on an island. Okay, but where this floor is set by this new I bar, which itself can be a function of time. Okay, so are the questions on the basic model, the basic phenomenology? Okay, so a question here, how much does the floor fluctuate based on how many islands are? Okay, so this is a very good question. One of the things we're clearly going to want to understand is this fluctuations of the floor with a large, but finite number of islands. 10 is not that big. And of course, if this goes down far enough, then we can get extinctions as we did here. Here, the total population across all the islands, which was the sum of all these, that went down low enough that I had extinctions. So one of the things we would like to ask about is about when do we have global extinctions, and when do those global extinctions, how do they depend on the number of islands and other things. So that's one of the things we need to understand. And that's one of the things I say, which the understanding of came later, and really with the sort of back and forth of the theory and the simulations. Okay, so now I'm going to talk about the theory, and let's see how much time left. Maybe 10 minutes before. Well, no, I should probably stop now and take questions. Let me just write down one thing, which is just sort of to lead as to where we're going to go. So this is now general method, which is dynamical mean field theory. It was developed in a lot in the context of spin glasses. And what we're going to do is we're going to focus on one island. Okay. And the dynamics of this is going to be coming from something that looks like noise from the others. Okay. But this noise from the others is going to be determined by the dynamics of all the others. So this is coming from all the others. So I need to understand this because, of course, this comes from all of the others, all other types on that island, and the new eyes on all of the other islands. So this is again affected by migration. This is going to affect it via the interactions. So it's coming from all these, and so determining what this noise is a big challenge, and one has to do that self-consistently. So this noise then has to be determined self-consistently. consistently. Okay. So this strategy is exactly like mean field theory for a magnet. It's exactly in the same spirit as that. There, one assumes that one spin, one makes the approximation that one spin here has an effect of a field coming from all the others, magnetic field coming from all the others. That magnetic field depends on the magnetizations of all the others. The magnetization will give you this field that gives this magnetization. You don't have to average this to give dynamic to this spin. We have to average that to get the magnetization. So it's exactly the same spirit as doing mean field theory for a magnet, and it's going to be valid in the limit that k is very large. So k is going to infinity. And when we do the things with the islands, we're also going to initially want to take I to infinity. And then, of course, we have to ask the crucial thing is what happens if those aren't infinite. Okay, so this will be the spirit, and I'll start next time with explaining really in detail how one does this and trying to explain the results that we've got. And then I'll follow that with things on more open questions and talking about evolution and things.
Okay, thank you very much. So other questions?
Hey, Daniel, I have a small technical question. So this might be silly, but does the fixed point correspond to the dynamical mean at all? Is that what's going on?
Okay, so when I'm in the antisymmetric model, okay, so the antisymmetric model where there's a fixed point, okay, so here in this antisymmetric model, this has a fixed point associated with it, and that, as Stefano worked out, theta equals 0 is the fixed point. Here, when I've got positive theta, in general, the average of the new I, and this is averaged over time, the time average of that will be equal to the fixed point value. Okay, that's true here. When I have migration, it's not going to be true. And when I've got migration, it's not going to be true because I can't straightforwardly average things. So where that came from was dividing this by this and averaging the LIs, the logs. And if you average the logs, this averages out and you get the fixed point condition. But you notice here, if I average the logs, then I have to pull out a 1 over nu I down here, and it becomes nonlinear, becomes extra nonlinear. So this is not true with the migration. Okay, so that statement is only true with without migration. As soon as I've got my migration, then there doesn't correspond to that. And in fact, if I look at the dynamics here, when I'm looking at all these distinctions, there's no fixed point. There's no stable fixed point in this case. Okay, there is a possible behavior of all the islands being in sync. If all the islands are in sync, then it's exactly like one island, but then it'll just fluctuate wildly and drive the extinctions like this. Okay, but the chaos will tend to go a little bit of differences will tend to make the chaos go asynchronous between the islands, and this crucial bit of the chaos desynchronizing, which is what causes this enables it to persist. Okay? So it's no longer true here that there is a fixed point, even a stable fixed point, and each island is doing sort of its own thing, but they're coupled to each other via the migration.
So we have another question from Miguel and then Mercedes. Miguel Rodriguez.
Yeah, thank you, Daniel. That was really cool. I have a question that doesn't really affect much of the math of the development of this model, but it's mostly on the biological assumptions of the phase interaction. So in this matrix that you use, you say that by definition, the interaction between phage and bacteria is basically an interaction of parasitism.
Right.
But we know that if we consider the prophage phase of the viruses, actually many viruses have a mutualistic interaction with the bacteria, and more recent work by Jed Furman, for example, shows that in the wild this is the norm, not the exception. How will that affect the stability of the
Okay, so as in everything with biology, there are all kinds of complications, and I'm not claiming at all here to understand specific biological systems. Okay, however, I would make the following statement: if all phages were prophages that had symbiotic interactions and they were not parasitic, they didn't attack. Okay, I personally think there would be a hell of a lot less bacterial diversity or phage diversity. Those behaving differently, then you can get stability and so on, and that stability can be there, but it will pay for some of the phages to adopt a different lifestyle, and then those are the ones that will drive this kind of dynamics, or can, and will drive, I think, the longer term evolution. Okay, so and this is just an instinct at this stage, but it's sort of based on developing understanding, is that really the absolutely crucial thing for diversity and for longer term evolution is really these sort of antagonistic interactions. Okay, one certainly can get a lot of diversity and complications coming from interactions via resources and so on. I think one has to cook things up much more to do that. And what I'm trying to convince you here is that something can happen with very little assumptions, right? So I made very specifically, I'm making assumptions here that I don't have the niche interactions. Okay, I don't have niche interactions. I'm not assuming anything special to interaction with itself. Now, the analogous thing for the niche, for the phage, would be to have specialist phages. Each phage is attached to one bacteria, okay? And then those will interact with each other. Then they'll tend to have cycles, or can have cycles, or can be stable. Again, there can be pressures for the phage to start doing something something different. But there you're assuming, in some sense, the answer. You're assuming that everything has a niche. And if I have a slight variant of the bacteria, a slight variant of the phage, that will no longer be exactly in that niche. And then you can get back into these kinds of situations. So I think that's a really important question. I hope that'll be something which will come up in the roundtable discussion next week. But I should be clear, I'm really trying to ask about which things, not surprising. If we can get things in really simple models, then we can say, geez, maybe they're not so surprising that we see them in nature. But that doesn't mean that we can apply it to particular biological systems, and there are always very large numbers of extra complications. The same is true in physics, I should say. I'm a condensed matter physicist, not an atomic or particle physicist. So I've always dealt with complications, and one has made tremendous progress by saying, okay, we're going to try to ignore a lot of the complications, look at simple models, and then we can sort of add the complications in one by one. And I think that really would be the goal here. In particular, I can ask, okay, maybe the way the evolution goes is that I drive things which go in the direction of they get more niche-like. That's a possibility. The other one is this assumption here, which is that I've said, I'm going to assume these are very small. I could say what's going to happen is there's going to be generalists that evolve. What does a generalist correspond to? A generalist corresponds to its S getting larger, right? It's doing well against all of the others. So the S's evolving would correspond to generalists. And I'm going to say something briefly tomorrow about that possibility, what happened in evolution. Okay. So the crucial thing here is we really shouldn't be talking about assembled models. We have to ask, can this evolve? Right? Of course, if it evolved elsewhere in the world, it can come together, assembled, and that can be relevant, but then it will continue to evolve. Bacteria and phages evolve fast, especially when they're in new conditions, like being with new 23rd cousins, instead of being with their close relative. So the crucial thing here really is to ask whether you can get to these kinds of things from evolution, with, again, reasonably simple assumptions about the evolutionary process. So that's what I'm going to sort of end with tomorrow.
So we have Mercedes, then please, Mercedes.
Yes, quickly. You just said that the important question about can you assemble it with evolution. So it was a bit my comment, but in reality, I think a lot of it will be very enlightening to connect these kinds of analysis to evolution on trait space, because where you connect the VIJs to the VIJs, it's not just evolving particular parameters, is that the result of that evolution gives structure to the VIJ.
Yes, so my last topic here tomorrow is to talk about phenotype models. So that's exactly going to be evolution in trait space. And I have some preliminary things to say about that. So what this would be where the VIJs, of course, are determined by the traits of strain I and strain J. And if I is a phage and J is a bacteria, those are really the traits associated with interaction. So I'm going to say a little bit about that at the end. So that's absolutely crucial.
I think it is crucial, and I think it is the challenge, because I think like it will also enable the empirical problem, which is if we look at some distributions, when you say this could look like neutrality, right? In reality, we have to ask what are these macroscopic properties that differentiate from neutrality and that tell us something insightful about the processes.
So I'm going to say something as a very brief and very preliminary, very conjectural still about the simple phenotype models in the context of the bacteriophage phage system and how that connects or might connect to what I'm talking about. Okay. So I'm going to say the things which I've just shown here today, those are all solid. There's one paper on those with a quite long paper, and the methods I'll talk about tomorrow. After that, everything is very conjectural, very much work in process, and particularly the continual work of Michael with Michael Pierce. And really, these are exactly the questions that I want to come to. So thank you for advertising my my talk tomorrow, which I guess is the last of 6 talks tomorrow or something, or is that today? It's the so thank you for being here, those of you that have survived the earlier ones.
Yeah. So we have one question in the chat from do you have an idea of how experimentally test chaos?
Yes. Okay. So one of the crucial things from here is that snapshots of abundance distributions can be very misleading. And I showed that just with this simple, idealized antisymmetric model, you got snapshots that look very close to a neutral model. Okay. You put in numbers and it just doesn't make sense for microbes. Okay. And that's also true with this state, which I've been talking about, where I've got many islands and so on. Again, if you take snapshots on one island, you will see things that look neutral. So the crucial thing is to look at the dynamics. The crucial thing is to look at the dynamics. You have to follow the dynamics of the strains with time. Now, this has been done, particularly in planktonic systems. Forrest Rohrer and others have done that and looked at that, and you tend to see chaotic dynamics of things coming up and down. Okay, that's a more complicated system, but the planktonic systems are just the kind of ones I want to think of in this context. They're much simpler than things like human guts, and they things mix together. They compete. The spatial structure things can move around, but really the secrets are in the dynamics. Now, in the long run, they're in the evolutionary dynamics as well as the ecological dynamics, and so you want to understand the sort of relations between the strains and so on. And you'd really like to be able to track the genes that were responsible for the traits that dominated the interactions. So coming back to Mercedes' points. So you'd really like to identify those and track those genes so that even if those genes were not always in the same organism, they were moving around, you could track the dynamics of those genes. Okay, that were associated, say, with the phage receptor and the tail of the phage that binds to that. It's a simple example. So really, one would like to be able to identify those and track those, and then you can use all genetic tricks to be able to track those. Okay, so that's really thinking about the sort of future and how one might hope to make contact with reality, but contact on sort of the conceptual ideas. And this is sort of a scenario for getting diversity and stabilizing and evolving diversity. It's not something which is think of a predictive theory in any detailed sense, but it really is a scenario, and it suggests what things to look at.
So let's take one last question from Almon.
Daniel, so I've seen a lot of work gone into using second quantization. Using, say, like what's only been done in spatial settings in geography. I was wondering, especially in this context of DMFT, if there's any real advantage or if it's just something purely aesthetic, because you say that for example, well, from what I understand of DMFT, you need small fluctuations around the average, but then again, I think would having a second quantized form allow a little more room
you can write things. You can write things as a field theory. You can start with writing a field theory for the dynamics, and that's a useful way to sort of derive things. Okay, but I want to make a comment about this because people coming from physics, as I do, one tends to like to put things into a form that one can then beat on and use the standard tools. Okay, some of those here in this particular context one can do, and these ideas that came from spin glasses and so on. Okay, And some of that you can say do with field theoretic. It turns out doesn't help much. You can just sort of check that you're doing things in a consistent way. There's another and much simpler problem, which I've worked on a lot, which is trying to understand dynamically, continually generated diversity coming from evolution in large populations. And this is say large bacterial populations in the lab, or viruses within a person, within a host, and they just evolve. They're racing the whole time against each other. This continual evolution. Okay, for that you can quickly write down asexual evolution looks like a field theory. I have never seen anybody get anything useful from the fact that it looks like a field theory. Okay. The things that I find I bring from statistical mechanics are some of the conceptual things and some of the ways of sort of thinking about asymptotics of how to approach problems and how to ask about questions robustness to convince oneself, or try at least, that what one is doing is not very, very special. Okay, so a lot of the things you can't do by sort of cranking out the methods that one has from statistical physics or from field theory, which makes it very, makes it a lot of fun, makes it hard. You can't just assign a problem saying, okay, here, go do this, and have a means of doing it. This is still very much, and the things I'm going to talk about here, to some extent, is still very much an art. And it's not sort of methods that you can directly take to be able to do it. You can write down things like this dynamical mean field theory, but you just can't get anywhere with it without a sort of lot of extra, conceptual ideas and mathematical, to some extent, trickery when one's only used it once. But one hopes that becomes a method.
Okay, so thank you very much. I think we can stop here, and we'll come back for tomorrow. We start again. So tomorrow is going to be a long day. We start at 12 Europe time, and we will end up again with Danny Fisher. So thank you very much.
Okay.
See you tomorrow.
Thank you. Thank you.
Thank you.
Me.